नको ना नको
ऑप्शन येत असते ते ऑप्शन वर क्लिक केलं की आवाज येतो.
गायकवाड सर तुम्हाला आवाज ऐकायला येतोय?
जरा बघा.
गायकवाड माझा आवाज येतो ना?
हो येतो येतो येतो.
करूया चालू गायकवाड सर?
हा येतोय सर, चालू करा.
Department of Chemistry, in collaboration with IQAC, organizes an international e-conference on Proteomics application to Biomedical Research 2022, that is PABR-2022, with the Department of Chemistry and IQAC Vignan Mahavidyalay Sangola, Taluka Solapur, Maharashtra, welcomes you for today's international e-conference on proteomics application to biomedical research, that is PABR-2022. The IQAC coordinator is Dr. Sade, the assistant coordinator of IQAC and anchor, myself, the convener and head, Department of Chemistry, Mr. R.S. Langwal sir. The organizing secretary is Dr. S.M. Kadam. The treasurer is Mr. N. Members of organizing committee, Mr. A.R. Ingavle, Dr. S.R. Shinde, Dr. R.B. Bhosle, Dr. B.S. Daune, Dr. S.B. Patwari, and Dr. V.P. Ubale. The organizer and acting principal, Dr. R.A. Phule sir, have taken efforts to make this e-conference successful, along with the entire teaching and non-teaching staff of the college. The Department of Chemistry is one of the leading departments, having 4 permanent faculty members. Right now, 28 graduate batches has completed their BSc course from this department. Currently, faculty members are doing their research in the field of supercapacitors, gas sensor, solar cells, etc. Department of Chemistry, in collaboration with IQAC, is going to conduct PABR 2022 2022 via Zoom platform. We are starting with inaugural session, and the resource person is Professor G.B. Zore sir, School of Chemical Sciences, Swami Ramanand Tirth, Marathwada University, Nanded, and president is Dr. R.A. Phule, acting principal of our college. So I request Head, Department of Chemistry, Professor R.S. Gaikwad sir, for introductory speech and welcome address. Mr. R.S. Gaikwad sir, Head, Department of Chemistry, is going to deliver the introductory speech. Gaikwad sir, it's over to you.
Gaikwad sir, please unmute yourself.
Good afternoon, everybody. The President of Inaugural Function, Acting Principal of Vidyan Mahavidyalaya Sangola, Chief Guest of Conference, Dr. G.B. Zore, and participants of the conference. I am immensely happy to welcome you to in conference held by Vidyan Mahavidyalaya Sangola to the International Conference on Proteomics Applications to Biomedical Research 2022, that is PABR-2022. I cordially welcome all the delegates and participants. I hope that this international e-conference on proteomic applications to biomedical research will enhance the knowledge regarding topics. It's our pleasure to have such a feast of knowledge in concern with branch of proteomics and related biomedical research. Thank you very much.
Thank you very much, sir. Now, I request IQSC coordinator, Dr. S.S. Dasade sir, for introduction about the today's chief guest, Professor G.B. Zore sir. Dr. S.S. Dasade sir.
Thank you, sir. The President of Inaugural Session, Acting Principal of Vidyan Mahavidyalaya Sangola, Chief Guest of the Conference and Guest of Honor, Dr. G.B. Zore, foreign invitees and participants of the conference, my dear friends, ladies and gentlemen, I am immensely happy to welcome you to e-conference and to Vidyan Mahavidyalaya Sangola to the International Conference on Proteomics Application to Biomedical Research 2022. At the outset, let me record my gratitude to Directors of Sangola Taluka Shetkari Shikshan Prasarak Mandal and especially to the Honorable President and all the directors for planning this type of conference. I am thankful to my colleague in the college for entrusting the responsibility of organizing the conference to me and Professor R.S. Gaikwad. Our aim in organizing the national or international conference or the event is to motivate and promote the student and research scholars, especially from villages. Dear friends, we have made every possible efforts to make this type of platform better and comfortable. Should there be any problem whatsoever, you can approach me directly. I extend warm welcome to one and all and wish you very pleasant and fruitful with this digital platform with us. Now I would like to introduce Raman Fellow and Chief Guest of the inaugural function of this international event, Dr. G.B. Zore sir. It is my great pleasure to introduce Dr. G.B. Zore sir. He did his MSc and PhD in Swami Ramanand Tirth, Marathwada University, Nanded. He is working as assistant professor and Raman Fellow in research lab, School of Life Sciences, Swami Ramanand Tirth, Marathwada University, Nanded, since 2006. He studied biotechnology, microbiology, botany, zoology. His areas of expertise is in the field of genetics and micromolecular biology. Also, he is expert in biotechnology. His research interest is in the field of Candida albicans of biology and plant biology. He did work on some prestigious research projects funded by central government and some foreign government also. He did work on ICMR-DHR International Fellowship of 27 lakh in year 2019. He did work on USA and BI-RAC of India, having the project cost of 65 lakh in the year 2022. He worked on Raman Fellowship, WSU Pullman, USA, in year 15 to 16. He also granted by SERB Young Scientist Scheme of 23 lakh in the year 2012 and 15. He got major research project of UGC of 15 lakh in the year 2010 and 13. He has also worked on UGC minor research project in year 2011 to 12. He worked on DST and Inspire Fellowship of 25 lakh in the year of 2012 and 19. The great personality Dr. G.B. Zore sir, he worked on UGC JRF for international student of 5 lakh in year 2014 to 19. Dr. G.B. Zore is honored by ICMR-DHR International Fellowship of 1920. He is also honored by Raman Fellowship for Postdoctoral Research 2014 to 15. He is honored by SERB FT System 2012. And one of the prestigious things regarding Dr. G.B. Zore sir is that he is nominated for Shanti Swarup Bhatnagar Prize in year 2019. Now he is guiding 6 PhD students and 2 MPhil students. He has published 18 research papers in well-reputed journals. His citation index, his H index is 9 and item index is 8. His article got cited 700 times. He is having 1 US provisional patent. He has organized 2 international, 3 national and 3 state level events. He got the membership of some prestigious association. He got membership of American Association of Advanced of Science USA 2015 to 18. American Society of Microbiology USA 2015 to 18. He is member of Society of Biological Chemists in the year 2004 to 5. He was also member of Indian Society of Mycopatho India in year 2004 to 7. He is also member of Moving Academy of Medicine and Biomedicine Pune in the year 2006 to 9. He got collaborated with Brown University, Providence, USA. He also did collaborated work with Washington State University, Pullman, USA. He also collaborated with Kanna State University, Manhattan, USA. He did collaborate with his work CSIR and NCL Pune. He has collaborated with Department of Biotechnology, University of Pune. He also did work with Punjab Agri University, Ludhiana, GB Pant University of Agri and Technical Patnagar, Chaudhary Charan Singh University, Meerut. He collaborated with Dr. Rajendra Prasad Central Agri University, Bihar. He did collaborative work with Jawaharlal Nehru Krishi Vidyapith. He also did work with Rajasthan Agri Research Institute, Jaipur, and one of the Meta Helix Private Limited, Bangalore. And his potential collaborators are Dr. Julian from London and Dr. Gill is from Madrid. He got many administrative responsibilities in Swami Ramanand Tirth Marathwada University, Nanded. We are here with this honored personality on this digital platform. I welcome him and all of you. Thank you sir so much. With this, I invite Dr. G. Bizore to inaugurate this international event by keynote address. And I am going to hand over this session to Dr. Jagannath Homre. Thank you, sir.
Thank you very much, sir. Now I request our resource person, Professor G.B. Zore, sir, for keynote address.
Thank you.
Professor G.B. Zore, sir, it's over to you.
Thank you very much. Thanks for the opportunity and thanks for the honor. I am overwhelmed by the words, kind words, and the opportunity organizing team has provided to me. And this is really, I think, one of the best opportunity I ever had to interact with the students and faculty member. And this is one of the best platform, this e-conference on proteomics, applications to biomedical research 2022. I would like to congratulate the organizer for organizing such a conference on nice topic, and because this biomedical research and biomedical applications has become so relevant, right? So since from last 2 years, because we are facing, not just we, whole world is facing this pandemic of COVID-19, and we know how we were actually considering ourselves that so advanced that we were neglecting the nature, we were neglecting everything, we were become more confident, but a small virus has shown us the position, or shown us where exactly we are belonging and how we should be careful about all these things like nature, and we should take care of everything, and we should learn how to live, right, sustainably, without damaging the environment, without damaging the nature. And with this, once again, I am thankful to the organizing committee of this particular conference. This college, Kadam sir has called me 2 days ago and asked me, sir, you have to deliver a keynote address for this particular conference. And I was actually busy because we were having another international conference going on in our school, in the university. So till yesterday, 11:30, I had guests and I was busy with those people. I didn't get enough time to prepare slides and all these things. But yes, whatever I have prepared, I would like to share with you people. And how much time I have for this presentation?
So maybe 45 minutes.
All right, so that would be more than enough for me. Yeah. So yes, so I'll be talking on proteomics, brief, right? Tools for understanding the disease biology, and what exactly is the proteomics, how we can use, how we can make use of this particular tool or technique, right? To understand how these diseases develop, whether it is infectious diseases or non-infectious diseases, because we are trying to develop diagnostics, trying to develop treatment strategies, right, for these diseases. But in order to develop the what we say host-friendly treatment and diagnostic strategy, first requirement or first prerequisite is to understand the disease biology. What are the causal agents? How exactly it is causing the disease? What are the mechanisms? And where are the opportunities? How we can intervene those disease progression. And to understand all these things, right, we need different tools, right? Molecular tools or other whatever tools we have. We need to use these tools. We need to make use of these tools. And since the advent of these new tools like genomics, proteomics, or omics technology per se, we are able to understand how exactly these diseases progressing, and then we are able to intervene those pathogens or those diseases, and then we are trying to develop strategies against those diseases. So proteomics, my slides are visible.
Sir, slides are visible, but they are not moving.
Yeah. So I haven't
Yes,
started that yet. So I will start the slideshow. All right. So we'll start with this. What is proteomics? Now it is visible.
Yes, sir. Now it is visible.
Right. So proteomics is the study of protein expression, regulation, modification, and function in living system for understanding how living system use proteins. Because we know proteins are the building blocks of this cell and all these organisms. And these proteins, because of its role, right, it is called as building blocks. It plays very important role in cellular organization, cellular function, and these proteins, either they play structural role or enzymatic role, right? They are catalyzing. They are also called as this biological catalyst, that is those enzymes. And these enzymes and these structural proteins are the key components or building blocks of the cell. So any disease that develops, right, that is associated with some of these what we say misregulation of proteins or mutation in genes that translate into the defective protein, right? And not just defective protein, but expression level of that particular protein gets changed. It may be down-regulated or it may be up-regulated, and that makes all the difference. And that's how we have seen in COVID also. There are different proteins, they are upregulated, and they are inducing the this immune response, and then that immune response is causing the cytokine storm. And most of the people who died because of COVID, they died because of this cytokine storm. So that's how these proteins are playing very important role in different activities in the cell, like organization and function and survival of the cell. So proteins, so before going to the details of this particular protein, we need to understand the central dogma of life. So I hope most of the students, most of the people are aware about this particular central dogma of life, where this DNA is transcribed into RNA, and RNA is translated into protein. So that's how our genome that governs our phenome or phenotype. So whole functions, right, where whole phenotype is determined or governed by our genome. And this genome is nothing but the whole set of genes present in that particular cell. And that set of gene is transcribing, producing the transcriptome of that particular cell. And the transcriptome, that is, transcriptome is nothing but the total RNAs, right, present in that particular cell at any given time. So that transcriptome translates and gives us the proteome. So proteome is nothing but the whole set of proteins present in that particular cell. So that's how this proteome is associated with transcriptome and genome, and downstream it is responsible for the metabolome of the cell or organism, and then that metabolome decides our phenotype or phenome. So that's how this omics, right, from genomics, transcriptomics, proteomics, right, metabolomics to phenomics. So that's how this genome is governing our phenome through this proteome. So that's why these proteins are the functional units. They are responsible for structural organization as well as catalyzing all these metabolic processes occurring in the cell. And that's why, in case we want to understand the role of this, understand the biology of the disease, we can try to understand how that particular disease is evolved, what type of proteins are modulated, what type of proteins are defective. And that's why this proteomic will help us what type of proteins are expressing, where exactly they are expressing, at what extent they are expressing, what is the rate of protein, right, expression, and what is the half-life of protein, that is degradation, and what is the physiological level of that particular protein, and whether that level is maintained or changed. In addition to that, we can also understand through this proteomics how these proteins are modified, because we all know after translation, these proteins are modified post-translationally, mainly glycosylation, phosphorylation, right, acetylation, and there are many more modification occurs in those amino acids present in the polypeptides. And these modifications are so important that these modifications make that particular protein attain the functional conformation. So these proteins need to form that peculiar or that typical structure, and the structure is formed because of this post-translational modification. And any change in this amino acid sequence or any change in this post-translational modification will affect the formation of that protein structure. And if that protein structure is not formed properly, right, if that protein has not achieved the proper conformation, that protein is going to be defective. So we can understand post-translational modification also using this particular proteomics approach. Then we can also track how these proteins are moving from one organelle, right, like endoplasmic reticulum to Golgi complex, and then how it is exported outside the cell or transported to different organelles in the cell. In addition to that, we can understand, we will also learn how these proteins are regulating metabolic processes, like whatever nutrients we are eating, like sugar. This sugar is providing, we call it as energy molecule, and this sugar, right, specifically glucose, is hydrolyzed or digested, right, and that process is called as glycolysis, and that involves several proteins, right, and these several proteins are breaking down that sugar, right, and this, during this breakdown, whatever energy stored in those bonds will be used by the cell for synthesis of ATP, and these ATPs are known as the energy currency of the living organism or cells. And that's how this energy, this breaking down or making the new molecules in the cell, that is called as metabolic pathways. These metabolic pathways are regulated by these proteins or enzymes. And any change, right, in the level of that particular protein or structure and function of this particular protein is going to affect the metabolic processes it is governing. And any defect in this particular metabolic processes will lead to the metabolic disorders. So that's how we can understand how exactly these proteins are modulated in these metabolic disorders, and then how we can fix it. In addition to that, we can also learn how this protein interaction occur, and that protein is important for this all the activities these proteins are carrying out. So that's how these different proteomic approaches, right, help us in understanding the biology. So it is not just disease biology, but whole, right? Like if we use this particular proteome, we can go for structural proteomics. So this structural proteomics will help us understanding how these organelles are made, what type of composition it has, right? Then how we can isolate those protein, and then these protein complexes, how these proteins are involved in the organization of this particular cellular structure, including organelles. Functional proteomics will tell us the exact function of different genes, and then any defective genes, and role of this particular proteins, that we will understand through this functional proteomics, how exactly the protein is carrying out this particular function, whether it is enzyme or whether it is structural protein. Proteome mining will help us in the drug discovery, right? Identification and validation of the drug target. Protein expression profiling help us in medical microbiology, understanding the signaling pathways, signaling mechanism, signal transduction, and disease mechanism. Post-translational modification, as I said, glycosylation, phosphorylation, and proteolysis, everything about that particular protein, how it is modified, how long it stays, what is the half-life, how it is going to be recycled, that we understand through this particular approach, that is post-translational modification, and then protein-protein interaction that we were talking about. So that's how these proteomics will help us understand total or all the aspects of this particular cellular organization, cellular functions, cellular metabolism. So it can help us what type of proteins are interacting, right? For example, this tumor suppressor protein. So we can understand how this particular tumor suppressor protein is interacting with other proteins and how it is suppressing the tumor formation, or how it stops the tumor formation, right? We can also understand the location of these different proteins, where exactly those proteins are located, and what are the proteins involved in different processes, like as I said, different metabolic pathways, different what we say mechanism like circadian rhythm, how it is regulated, then homeotic gene expression, different processes like embryonic development, and all these things we can understand using this proteomic approach. And it is because we would like to understand these processes, so that once we understand that those processes, right, we can definitely intervene whenever there is some problem, whenever there is some modulation. So we can fix it easily. We can understand, we can diagnose it easily. So that's why we need to understand this particular disease biology. And proteomics is one of the most valued, most reliable tool for understanding that. Because after the sequence, right, Human Genome Project is completed in 2001, 2002, that draft of whole genome was published, but people were confused. What does it mean? So they reported almost 25 to 30,000 genes in human genome, but then how those genes are functioning, right? How those genes are regulating this whole physiology or whole processes in human cells was not known. So we were just knowing it is just like a draft, and we were not knowing how this particular genome is significant. And just to understand the significance of this particular genome, we moved further towards the new tools and technologies, like we started understanding expression of those genome, and then we also started understanding how exactly this expression product of the genome, that is proteins, functions. And then understanding one or two proteins was not enough to make some sense out of that particular whole genome sequence. And that's this concept of proteome has came into existence. And I'm proud that one of our students from New Arts College, now he is a PhD, Dr. Gajanan Sate, he worked and they have provided the first draft of human proteome. So that was a big news and that was a big story that was published in Nature, I think a few years ago, I think 5-7 years ago, they published that paper. And that's how our people, right from Maharashtra, they are contributing in this particular tool, and they are trying to make this particular understanding more clear. So this is basics of proteins. I think most of these people are from chemistry background, so you are more familiar with this particular structure. So basics of protein. So it is a polymer of amino acids. We know there are 20 naturally occurring amino acids, and these amino acids are having N terminal and C terminal, right? And then they are going to have those other groups, and based on that groups, there are total 20 different types of naturally occurring amino acids that is forming or polymerization of this particular amino acids will form the polypeptide. And once these polypeptides are modified, transposed translationally, they will form the proteins. So as I was talking about N terminus, C terminus, and you can see there are different structures, right? Primary structure, it is just based on the formation of polypeptide bonds between two amino acids, and then polymerization of these peptides will give you a polypeptide, and then secondary structure will form the either it will be forming the alpha helices or beta sheets, and then that will be converted into a tertiary structure through the non-covalent interactions, right? And that will be forming the tertiary structure of the protein. And then these proteins, right, different subunits come together forming the quaternary structure in case of multimeric complexes, and then that protein becomes functional. So any defect during this particular process, right from attaining the primary structure through these polypeptide bonds, or secondary structure through this non-covalent interaction, right, forming alpha and helices and beta sheets, or tertiary structure, it will affect the function of that particular protein. So that's why this understanding basics of this particular protein structure is very, very important. And that's how this proteomic tool is helping us understanding that particular protein structure. So this is the basic strategy for protein identification. So we can separate protein. I hope you know that you can separate this. You can prepare protein from those tissue or cells you are interested in, right? Analyze those cells, isolate protein, and then separate those proteins on the this polyacrylamide gel. Then you look for those protein, right? Protein band of your interest. If you are interested in specific protein, you can just get that particular protein presented from the gel, right? Digest it into the peptides, right? And then you can go for mass spectrometry, that is peptide mass fingerprinting or peptide sequencing. And then once you got the peptide mass or peptide sequence available, just compare it with the databases. And that database comparison of that particular data, whether it is mass fingerprinting data, peptide mass data, or peptide sequence, will tell you what type of protein it is, what is the protein this thing name, and then once you identify that particular protein, you will see if there is any change or any modification. So that's how this is already discussed. So we can understand the location, we can understand the level or abundance of that particular protein, and then different types of modification, their activity, and how this protein interact through this proteomics approach. So these were the conventional approaches, like we from this one-dimensional gel to this 2D gel electrophoresis came into existence, that is called as isoelectric focusing, where these proteins initially separated based on the mass, right? And then in this second dimension, it is separated based on the charge. So that's how we can separate those proteins in 2 dimensions based on the mass and this charge, and then it will be easier to separate or isolate that protein of our interest, and then we can look for the details of that particular protein through mass spectrometry for the identification of that protein. So this is how these proteins then digested into peptides, and then peptides are subjected for this what we say mass spectrometer. They are producing different these ions, right? And then you can select the ion of your interest. You can further, that ion means that is a peptide. You can further digest that peptide and go for another mass spec analysis, and that's how that mass spec will give you different fragments, and those fragments based on the mass by charge ratio, these fragments are separated and identified. So based on the mass and charge of that particular fragment, you can compare it with the databases and see what type of protein that particular peptide is belonging. And that's how you can identify that particular protein. So there are different approaches, bottom up and top down approaches. You can identify those proteins. Sometimes you use these chromatographies to separate those proteins. Then once you separate those proteins, digestion, and then those peptide you can subject to the mass spectrometry. And after you have this mass data, you can. All right, so we will come to that. After once you have that mass data, you can compare that mass data in the databases. So this is the approach people are using, targeted proteomics, to understand the proteins associated with different tissues, different organs, and different diseases. So identification of these proteins, we need to understand the level or expression level of that particular protein, at what concentration that protein is present in the cell, and what is the physiological concentration of any protein. So this quantification of this protein is carried out, right, using different strategies like this ICAT, isotope coded affinity tag, where we use 2 different types of tags. We label our control sample with one tag, and then our test sample with the heavy tag, and then mix it, and then subject it for the mass analysis. In between, we fragment those protein. After labeling, we digest, right? Digestion के बाद we label it, and after labeling, we analyze the mass of those particular fragments, and that fragment, based on these 2 tags we are using, we will come to know the level of that particular protein in control and in test samples. In addition to ICAT, there is another type of tag called isobaric tags for relative and absolute quantification of the proteins. So this is how this mass spectrometry works. What happens? Sample, you just introduce the sample, and then that sample, that peptide sample, is ionized. So there are different types of ionization strategies, right? And once that particular peptide is ionized, right, and then that will be what we say, that will be moving towards the detector, and that this time required for that ions to reach to the detector, right, depends on the mass and charge of that particular peptide fragment. And that's how we separate those protein and then detect it through the detector. So this mass spec basically involves the production of these ions of these fragments or peptides, and then separating those fragments in the phase, right? And this separation occurs based on the mass and charge, mass by charge ratio of that particular fragment. So there are different ion sources that electrospore-ionization, like that, matrix assisted desorption ionization, and these analyzers are also different, like quadrupole, ion trap, time of flight, and then different detectors are being used in mass spectrometer. So based on your requirement, your application, and your this thing need, you can select the suitable machines, right? So like matrix assisted laser desorption ionization, that is MALDI-TOF, and then electrospore ionization, where Nobel Prize in 2002 in chemistry, Mr. Fenn and Dr. Tanaka, right? So both of these techniques, they won the Nobel Prize for this particular discovery. So this is what happens here in case of MALDI. So you just mix the sample with matrix and then expose that matrix to the laser pulses, and then which in response to these laser pulses, right, this will desorb and ionize those molecules, right? So matrix is a small organic compound with property of high absorption of laser, right? And then it prevents decomposition of the sample and donates proton for ionization. So high ion yield, non-destructive ionization of both large and small molecule. That's how once those ions are ready, it will be detected through the detector after this ion detectors are there. So this desorption will lead to the movement of those ions towards the detector. So another one is the electrospore ionization, where these ions are generated directly from the solution, right, by creating the fine spray of highly charged droplets in the presence of strong electric field. And then vaporization of these charged droplets resulting in production of singly or multiply charged ions around 200 Daltons, 2000 Daltons. And then multiply charged species are also possible. But generally, this EI is coupled with the ion trap or quadrupole, and then that's how analyzers will detect those ions through this based on the mass by charge ratio. This is quadrupole analyzer, where this ionization is carried out only using electric field, and that's how these ions developed through this particular quadrupole analyzer, it will be detected. So ion trap, I will not go much into details, but once you have those particular ions developed, and those you'll get different types of that mass spec data, right? Mass or peptide fingerprinting data, right? Or mass library that you compare with the databases like Uniprot, and then protein databases available, and then you identify based on the mass of those particular peptides, you identify what type of protein that particular then how many peptides of that particular protein are identified in that particular mass library, and what is the concentration, what is the level of that particular peptide. So that will tell us what is the level of that particular protein present in the control and test. And then once we identify that protein, we will look for the reactome, right, or interactome of that particular protein based on the curated evidence, and databases like people have already uploaded their findings and stored this information in the form of databases like PRIDE, and there are different databases like reactome, where you will find the different proteins and their interacting or reacting partners. Here you can see Uniprot data, where you will get all the data for the different reported proteins. This PRIDE archive, where we can upload whatever protein mass data we generate. Everyone is uploading and storing that data so that other people can use the database for their own studies. So interactome, you can study the interactome. So what type of proteins your identified protein is interacting, and what is the role of that particular protein. So there are different databases in addition to PRIDE, Interactome, and Reactome. These are some of the databases where you can find this data for peptide mass mapping and sequencing. With this, how this particular proteome is helping us understanding disease biology. Recently, they have identified different proteins, right, associated with cancer, and then that will be helping us to understand the main culprit associated with the, this particular cancer. People have also identified the how exactly this SARS, right, COVID is transmitting from vertically transmitting from mothers to the children or neonates. So likewise, these are different applications, and it is all going on continuously. So we are understanding this disease biology, whether it is infectious or non-infectious disease, and this proteomics approach is helping us in that particular. So with this, I would like to just talk in brief about what exactly we are doing in our lab and how we are using this particular tool in our research. So as we know that we work on Candida albicans, and we would like to understand particular organism, right? The biology of this particular organism, because it is an opportunistic pathogen and it is lethal, especially in immunocompromised patient. And as it is an eukaryotic organism, right, developing this treatment strategy for this particular organism, it is difficult compared to the bacterial pathogen, because being an eukaryotic cell, right, it mimics in many ways as our human cell, or it mimics the host cell, and that's why any target we identify or use for this drug, that is going to have very severe side effects in the host or in humans. So that's why it has become difficult to develop the new or novel strategy against this fungal pathogen, including Candida albicans. And recent increase in the drug-resistant isolates in the clinics, right, of this particular organism has increased the significance of clinical significance of this particular organism. Candida is this is organism present in the different morphological form, right? That is this cell, hyphal cell, pseudo-hyphal cell, opaque cell, or chlamydospore. So it exists in different morphological forms, and again, that makes it more difficult to detect or what we can say defend, right, by our defense system or our immune system. So many times our immune cells fail to identify or recognize this particular organism, right? And because these defense mechanisms are always detecting any invading antigen or any molecule, any cell that comes in our enters our body, it will be detected by these defense cells, immune cells, and it will be destroyed. So unless this immune cell identify and differentiates or identify it as a foreign body or invading body, this cell, this immune cell cannot destroy that cell. So identification just to hide from this defense cells or immune cells, Candida changes its morphology. And in addition to evading these immune responses, this change in morphology of this particular Candida albicans cells again provides it an advantage to survive it at the different microenvironments or different organs in the human body, and that's how it makes it difficult to treat organism, and that's how already limited number of these antifungal agents are failing against this particular pathogen, and that results in the death of patients, right? And in high number, basic kind. So emergence of resistance has again contributed significantly in this particular lethality, or what we can say fatality of this particular disease. So you can see almost 7 lakh deaths per year, right, are occurring because of this particular antimicrobial resistance among the infectious pathogens, whether it is micro, right, bacteria or fungi. But and then it is also increasing. That number is going to go up, and it is going to be almost 10 million deaths per year by 2050 if we are not identifying novel and potent antifungal or antimicrobial agents that is going to be a number one killer of the reason for the deaths of the patients. So how exactly this antimicrobial resistance emerges, and then what is the reason behind that, how we can take care of this particular drug resistance mechanisms. So just look at this. Because of this proteomic approach helped us in understanding the emergence of this drug resistance among the pathogens. So you know that all the antifungal agents, or most of these antibiotics presently available in the market, they are targeting either its cell wall or membrane of this particular pathogen. And as this cell wall and membranes are highly dynamic structure, it keeps changing. So if it is present in yeast phase and if it is present in the hypophyll cell, so composition of this cell wall and membrane changes, and that makes that particular target, right, drug target, whether that particular, for example, these or echinokindins or azoles are targeting the membrane of this particular fungal pathogens. So if it changes into different morphological form, the composition of that particular membrane, specifically sterol content and other this thing, it is changing. So the level of that particular antibiotic we are using, or dose of that antibiotic we are using, antifungal we are using, many times it becomes inappropriate, right, for killing that organism. And if we expose that organism to sublethal dose of the antibiotic, that leads to the emergence of antimicrobial resistance. And that's how this inappropriate targets, right, that currently available, right, these drugs are targeting, that is making this drug resistance more frequent, and that is actually facilitating the drug target. So what our plan is, so we are trying to identify targets, right, that will never change based on the this morphology or the site of infection of that pathogen. And that's what we are proposing. That is our focus of our study at the SRTM University. So I would just like to tell you the hypothesis we are working on. So our hypothesis says a process and or a molecule of a pathogen that remain constant irrespective of the niche as well as a patient-specific variation could be the ideal target for broad spectrum antimicrobial agents. And that's how, if we have a target that is not going to change, level of the target is not going to change, if we use such target, I think that will avoid this emergence of drug resistance among the pathogens. So our objective is understanding basis, right, of this morphological differences in kind albicans, and then some of these studies we have carried out. So this is how we are using this particular tool. We are trying to understand proteome of this kind albican cells present in different cellular forms, whether it is yeast, hyphae, pseudohyphae, opaque form, chlamydospore, and just trying to understand how these cells are different, or how the proteomic profile of this particular kind of albicans cells is different, and how we can use that information for developing novel and more potent antifungal agents, or identify targets that will help us developing the more potent antifungal agents. So this is the strategy. We are just getting those cells of different morphological form, lysing those cells, isolating or extracting proteins, and those fragmenting those proteins through trypsinization and subjecting it for the this liquid chromatography mass spectrometry that will give us the swath spectral library. So we are going to generate that spectral data, right, of the fragments we have separated, and then that spectral library will be subjected for identifying the proteins using protein databases available. And that's how we are also validating that proteomic data using real-time PCR, or then using cell cycle assay, or then flow cytometric assay. So what we did was we have identified different microenvironment responsive changes, or microenvironment responsive proteins and genes in the Candida albicans, right, in different morphological form, whether it is yeast, hyphae, pseudohyphae, right, chlamydospore, etc. And some of those papers are already published. I will discuss about some of those proteins, what is our finding. Another important finding of our lab was we have first time correlated this morphogenesis of Candida albicans with the oxidative phosphorylation. And this was the first report we reported in 2018. We have also reported first time, that is how this capillary hydrostatic pressure is inhibiting Candida albicans morphogenesis, right? And that is also a significant finding of our lab. In addition to that, we have also a fungal specific domain in the HMG co-reductase enzyme that is conserved right from lower eukaryote to humans. And this is how we have reported different this expression pattern of different proteins, right? That is ALS, or agglutinin-like sequence proteins, and lipases that is involved in the virulence of Candida albicans. In addition to that, we have completed sequence of the Candida albicans strain, whole genome sequence we prepared and is uploaded. And we have identified, based on this particular information, we have identified almost 12 plant-derived agents as potent inhibitors of Candida albicans growth. Not just that, we have also understood, we have also made an attempt to understand mechanism of action of those particular molecules, how those molecules are inhibiting Candida albicans growth. This is something I just wanted to show you how we understood that low hydrostatic pressure, that is low means that is non-significant hydrostatic pressure, can inhibit morphogenesis of Candida albicans. That was recently reported. That this paper we were talking about, that serum responsive proteome reveals correlation between oxidative phosphorylation and morphogenesis in Candida albicans, where we have correlated that. This is how we identified proteins at different temperature in different morphological form, and then we correlated those proteins. We annotated the proteins to different organelles and different metabolic processes, and just assign those functions to those particular proteins. So this is how we predicted that based on our proteomic study, we hypothesized that this is the cell wall, right? How this cell wall composition is changing, right, in different morphological form. So in hyphal and in yeast form, how this cell wall is different, and this difference in the cell wall is associated with differential responses of this particular cell towards the antifungal agents. And that's how this is another study we have published recently. So we have identified this proteome of kind albicans during chlamydospore formation. And then this is another paper we published, chlamydospore specific proteins of kind albicans. And this is what we found during chlamydospore formation. So you can see different processes, right? How those processes are affected. So you can see that right from signaling pathway. So what type of pathways are upregulated, what type of pathways are downregulated, that we identified based on the different protein. So level of different proteins. So our study showed that glycolysis, TCA cycle, glycolytic cycle, and fermentation has gone up during this chlamydospore formation, right? And then this cell wall composition, how it is changed. So some of the protein like GPI anchored protein were upregulated, and maximum regulation of almost 200-fold was found in one of the proteins, that is GPI anchored protein. Then mannal contained glucan, chitin, and then ergosterol, phospholipids, sphingolipids, fatty acids. So composition of this cell wall and membrane is again changed, and most of the proteins involved in the biosynthesis of these particular components were upregulated. You can see that almost 200, more than 200-fold expression, right? Over expression of some of the proteins in cell wall, 20%, 20-fold more expression of these proteins involved in this ergosterol biosynthesis was reported by us. Then, in addition to this cell wall and membrane composition and metabolic pathways, this stress tolerance mechanisms, right, or proteins involved in stress tolerance mechanism were also upregulated, because we have subjected those kind of, we can say, to a stress, and that stress is inducing the chlamydosporulation, because chlamydospores are considered as a resting body, and that is more dormant. That remains in the dormant stage, those stresses. So that's how this proteomic approach helped us in understanding the types of modulation in metabolism, modulation in structural organelles like cell wall and membrane, and then mechanisms that is helping that particular cell to tolerate the stress. So we could understand those things using this proteomic approach. You can see detailed these pathways, what type of proteins, right, or what type of enzymes are modulated, whether it is upregulated. Mainly those shown in blue, those enzymes are upregulated, and enzymes shown in red color are downregulated. So you can see different metabolic pathways, right from nucleotide metabolism, carbohydrate metabolism, glycolysis, fatty acid synthesis, TCA cycle, amino acid metabolism, etc. All those metabolisms are modulated during this morphogenesis, and this mass spec data helped us understanding the mechanism of this particular morphogenesis. So you can see here signaling pathways, that CMP pathway is modulated. Then this trihalose accumulation usually occur for the survival of those cells in response to stress condition. And then this different proteins involved in different metabolic processes are also modulated. So that is the model we propose based on our proteomic data. Okay, these are the changes occurring during this particular morphogenesis of the yeast cell to chlamydospore. In addition to that, omics approach, we have also identified different differences in the fatty acids during this morphogenesis, this low hydrostatic we already talked about. And then we actually working on some of the plant molecules, but once we identify those different plant molecules, we try to understand how exactly these plant molecules are killing or inhibiting growth of this particular kind albicans. So we use different proteomic approaches, and we understand those proteins, right, or piperin responsive proteins, and we found that what type of proteins are upregulated, what type of proteins are downregulated. So proteins involved in cell cycle, right, then translation, transport were downregulated, and that's how this modulation in proteins in response to piperin will affect the cell cycle, and then that induces the not just cell cycle. So this piperin is inducing the what we say this membrane cell wall and membrane damage that is producing reactive oxygen species, and that leads to the modulation in the cell cycle and finally apoptosis in Kendall Beacon cell. So this is how you can see this is control and test, right? The control is shown in this particular you can see this red strip, and then green it is shown as a test, and you can see how these protein are expressed differentially. So blue is down regulated, and then red or brown strips shows the overexpressed proteins. So this is how that proteomic data is validated using flow cytometry and cell cycle analysis. Similarly, we also identified how this limonene is inhibiting Kendall Beacon's growth, and that we based on our proteomic data, we propose this particular model that this limonene is interacting with the membrane or inducing the membrane damage, and there is membrane damage. So based on our proteomic data, we have shown what type of proteins are down-regulated, what type of proteins are up-regulated, and what is the significance of those proteins, how this protein, misregulation of this protein will affect the Kendall Beacon cell and how it is causing apoptosis in Kendall Beacon cells. So this is how we have understood mechanism of action of these different plant molecules. This is the mechanism of piperin, and this another side you can see mechanism of action of this menthol against Kendall Beacons, and that's how we have understood the mechanism of action of this particular different plant molecules, as well as the modulation of the proteins, right, during this morphogenesis of Kendall Beacons using this proteomic approach. So that's how this proteomics tool is helping us understanding the proteomic changes occurring during this disease development in host and even in pathogen, and that is very important. So understanding this particular modulation, right? Because this modulation will tell us the physiological modulations in that particular cell, whether it is host or whether it is pathogen, and that will provide us an opportunity to intervene with those physiological changes in the pathogen as well as in the host, and we can develop a strategy for diagnosis or for treatment using this particular proteomic approach. So with this, I would like to acknowledge the contribution of my lab members, all the postdocs, PhD student and MSc students who is actively involved in this particular study. We also would like to acknowledge support from the funding agency like ICMR, then UGC, then USAID, USA, and then DBT BIRAC, Government of India, and for the what we say funding, right? They have provided financial assistance under different schemes. We also like to thank different collaborators from different universities, national and international collaborators, for providing the technical support, providing the material support for this particular study. And with this, I would like to stop here and thank the organizer for your kind support and organize this opportunity to share our views on this proteomic tool in understanding this or application of this particular proteomic tool in the biomedical research. So thank you very much. I hope I stick to my time, and then pardon me if I have exceeded. So thank you very much.
Thank you, sir. Please, sir, stop the screen sharing.
Yeah.
Thank you, sir, for your remark on tools for understanding disease biology. Also, you have mentioned the types of proteomics and their applications to biology. Also, in your talk, you have mentioned how proteomics provide significant biological information for biological problems. Also, you have provided some online database for protein information. Then you have mentioned the concept of antimicrobial resistance. Thank you, sir. Now I request our Honorable Acting Principal and President of inaugural session, Dr. R.A. Phule sir, for his presidential address. I request Dr. Phule sir for presidential address.
Thank you, sir. Dr. Dhore sir,
Thank you,
for
sir.
the such information keynote address on Proteomics. The excellent and tremendous information is a very much helpful to enhance the knowledge in the concerned area of research. Good afternoon, sir. It's my great pleasure to welcome to all resource person, Dr. Atul Deshmukh sir, University of Copenhagen, Denmark, Dr. Diana Somodoa, University of Copenhagen, Denmark, Dr. Balaji Rokde, Lia Hinsley in Ireland, and Dr. K.D. Sonavne, Shivaji University, Kolhapur, Maharashtra. Participants, students, and faculty member, today we are here for the 2 days international e-conference on Proteomics application to biomedical research 2022, organized by our college chemistry department and internal quality assurance cell. I also welcome all the eminent speakers and guests for all over country and outside country. The chief patron of this conference are Mr. Ramchandra Khatkale, President Sangola Taluka Shetkari Shikshan Prasarak Mandal, Mr. Chandrakant Deshmukh, Director, Dr. Aniket Deshmukh, Dr. Mr. Vittal Rao Shinde, Secretary Sangola Taluka Shetkari Shikshan Prasarak Mandal, then member Baban Rao Jankar, Dr. Ashok Rao Shinde, Dr. Pradhyapak Deepak Khatkale, Jayant Jankar, Mr. Avdut Umtekar. I would to congratulate the organization committee of this international e-conference, Mr. R.S. Gaikwad, convener and head of department chemistry, Dr. S.S. Dasade, IQSC coordinator, Dr. Ken Kadam, organizing secretary, Mr. Adling ANP, Mr. A.R. Ingole, Dr. S.R. Sinde, member organizations such as conference, Dr. B.R. Bosle, Dr. B.N. Devkate, Dr. Purwari, Dr. Ubale. This is our first international conference in the academic year 21-22. This completely connected on the digital platform due to COVID-19 pandemic. Vigyan Mahavidyalaya Sangola is established in 1991 by the Sangola Taluka Shetkari Shikshan Prasarak Mandal by the late Dr. Ganpat Raoji Deshmukh ML with the motto Vidya Savarthi Shadiye. The college has the science, arts, and computer science faculties. There are 17 UG departmental and 3rd postgraduate with the 1700 students. The college is affiliated Punyashlok Ahilya Devi Holkar Solapur University, Solapur. The college is located in Western Maharashtra in the Dotponi area. The college has a 75+22 teaching and non-teaching faculties. The college is reaccredited by the NAC Bangalore. The college is one of the best college in the university. The college is a very rich library with implemented services. The college focus on the student skill, other job oriented and short term courses by all the department. The college has a NSS unit which placed many students in the police, military, CRP and banking. The college has 60 plus classroom with a DLP, 4 computer labs, 7 science labs, gymkhana, language labs. The college has 15 kV watt solar system to generate own electricity. For the student, the rural, especially to the girls, through it is co-educational college for the empowerment of the girls in the rural and the drought prone area. With the intense management has struggled for that, and it say to some extent the I and May college colleagues they get succeed in it. Younger generation in the properties of the country, and I should be multidimensional, should not remain away from the main development stream, even through they are economically and socially from the weaker section. Keeping this in the mind, we always give the performance to such student at the time of admission. Later on, as per demand, we have also started computer-based courses to make them able to oriented. In future, we plan to start some PG courses so that the girl students can be completed their educational here only. Right now, we have BA, BSc, MA, ECS, MSc, and PhD program in the campus. The college proposed to start MSc Organic Chemistry from the next academic year. Thank you very much. And once again, I welcome to all. Thank you, sir.
Thank you, sir. Thank you, sir, for a presidential remark. You have mentioned the main aim of the conference. Also, you have focused on the history of the institute and current programs. So thank you. I request my senior colleague, Mr. A.R. Ingole, sir, ANO NCC unit, to pay vote of thanks. Mr. A.R. Ingole, sir, please pay the vote of thanks.
Now time comes to express vote of thanks for this session. First of all, we express our sincere thanks to Professor Dr. Gajanan Zhore of School of Science, SRTM University, Nanded, by giving a keynote address of our international e-conference on proteomic application to biomedical research 2022. In his keynote address, he told us about the proteomic as a tool for understanding biological diseases, as well as all details about the proteomics. He also told us about the structure and the function of DNA, RNA, proteins, etc., and how proteomics are useful for the animal bodies. Once again, we are thankful to Dr. Gajanan Zhore sir. Also, we are thankful to our sincere and very active principal of our institution, Dr. Raghunath Phule sir, for giving a presidential address about the conference to all the participants. We are also very thankful to IQAC coordinator, Dr. Shankar Dasade, as well as Dr. J.V. Thomre, for giving all types of support for organizing this conference. Our sincere thanks goes to all delegates, the resource persons, the chairpersons, the participants, and all persons who have directly and indirectly supporting for the success of this conference. Once again, on behalf of our institution, department, we are thankful to all of you. Thank you.
Thank you, sir. Thank you all of you for being with us. I declare with permission of organizer, the inaugural function is over. The next session, that is invited talk by Professor K.D. Sonone sir, will begin. Once again, I welcome all in second session. The session coordinator is Dr. S.N. Kadam sir. Dr. S.N. Kadam sir, it's over to you. Kadam sir, please unmute yourself.
Thank you, Dr. Thombre sir, for giving me accession of the second session. Myself, Dr. Kadam S.N. from the Department of Chemistry, Vigyan Mahavidyalaya, Sangola. I am here as a session coordinator of the second session for the International E-Conference on the Proteomics application to Biomedical Research 2022. I would like to introduce the chairperson of this today's session, second session, that is Professor S.B. Patwari sir, who is a HOD from LBS College, Dharmabad. He has a huge teaching experience of 13 years for UG and PG student. He had published up to 50 research papers in the reputed national and international journal. He had guided for 12 students for MPhil and PhD. He had also published 3 books in the concerned research area. He had also awarded 1 Indian patent for his research. Again, he have also completed 1 major research project granted by UGC. He had worked as a ex-BOS chairman of Swami Ramanand Tirtha Marathwada University, Nanded. He had now currently working as a member of Academic Council, Swami Ramanand Tirtha Marathwada University, Nanded. He is also working as a member of Faculty of Science and Technology, SRTOMI, Nanded. He is also a member of a PG Board of Education in Colleges, SRTOMI, Nanded. He is a chairman of Chemical Sciences, SRTOMI, Nanded. So, with this brief biodata of Dr. Patwari sir, I would like to hand over the session to today's chairperson, Professor S.B. Patwari sir.
हॅलो, आवाज येतो ना सर?
येतो. सर म्यूट झालं.
Dear sir, please unmute yourself.
Yes. Hello.
Now it is okay.
Good afternoon to all. Thank you very much for an introduction, Dr. Kadam sir. First, I congratulate to all management members of the institute, principal of the Department of Chemistry, head Department of Chemistry and his team, ICAC section, and all organizing committee members to organize this beautiful event. I would like to thank all these dignitaries to provide me a chance
as a chairperson for this beautiful event. Friends, we are very lucky to have chair resource person for this session, Dr. Sonone sir. I am very glad to introduce Dr. Sonone, Professor Dr. Kailas Dashnath Sonone, Associate Professor, Department of Microbiology, Shivaji University, Kolhapur. Sir did his MSc at Shivaji University, Kolhapur in 1996 and PhD from NCL Pune, Pune University, Pune in 2003. He has teaching experience about 16 years at PG level. Sir supervised 4 PhD students and still 7 students are working under him. On his credit, there are 39 research papers at national and international repute and about 119 conferences he has attended with paper publications. He has succeeded to organize 1 international and 4 national level conferences as a convener as well as organizing secretary. Sir got research projects from different funding agencies like DST, UGC. I'm very proud Sir got the grant of 32 lakhs funds from University Grant Commission, New Delhi. He received Senior Research Fellowship twice, CSIR, New Delhi, from 1999 to 2003 and 1996 to 1999. He also worked as a postdoctorate fellow at USA. Nature of the work, very interesting. Homology modeling, sequence analysis, molecular docking, and molecular dynamics stimulation studies on K+ ion channels. Apart from all these, sir is very active to participate different academic bodies of the universities. He is a BOS member, Biotechnology, Shivaji University, Kolhapur. BOS member, Biotechnology, Dr. Babasaheb Ambedkar University, Aurangabad. BOS member, Biotechnology, Swami Ramanand Tirth, Proteomics, Nanded. Sir is a life member of Association of Microbiologists of India, reviewer of several international peer-reviewed journals. In brief, I can say we are very lucky enough to have such personality with us. On behalf of organizing committee and the convener organizer, I am requesting to Professor Dr. K.D. Sonone sir to start his lecture.
Okay, thank you, sir. I hope I am audible to everyone. Hello.
Yes, sir.
Am I?
It's audible.
Okay,
Yes,
all right, all right. Hello, can you see my presentation?
Yes,
Yes, sir.
sir.
All right, okay. So first of all, I am very much thankful for the organizers for giving me this opportunity to talk in this particular conference on biomedical applications, Proteomics biomedical applications organized by Sangola Mahavidyalaya. So first of all, on the behalf of organizers, let us say as per the discussions, I have been given the topic kind of a proteomics approaches to understand some of the multi-drug microorganisms, particularly few bacteria and few fungus molecules. And this particular research that we do at Department of Microbiology, Department of Biochemistry of Shivaji University, Kolhapur. So let me begin with the particular the slide which we are seeing here. That is, what is the difference between traditional drug discovery and computer-aided drug discovery? So if we go by traditional drug discovery, so let us say we have to spend more than 14 to 15 years of time by understanding some of the processes, particularly to target identification, then target validation, then lead identification. So target means here a receptor or any protein or any enzyme that can be identified by using certain proteomics approaches, proteomics techniques. And once we identify the target or any receptor or any enzyme, which is responsible to cause any disease or responsible to cause the drug resistance, that antibiotic resistance, that would be our target, let us say. And lead is the compound drug. It could be antibiotic, it could be a secondary metabolite, or it could be a drug molecule. So that lead molecule can be identified. Then optimization of that lead molecule, that is called as the candidate optimization. Then one can go for preclinical studies. All right. And then after having a preclinical studies, one can go for clinical aspects or clinical studies, then phase 1, phase 2, phase 3. And whole this procedure takes nearly 15 years of time. And that pharmaceutical company who is doing this particular task needs to spend more than $800 million. So imagine 15 years of time and not spending more than $800 million to discover one drug molecule or to get that drug molecule into the market. But afterwards, what happens sometimes, FDA, that is Food and Drug Administration, they say that this drug creates some kind of a side effects. So they try to ban on that particular drug. So imagine what would be the situation of that particular drug molecule, that particular pharmaceutical company, all right, after spending 15 years of time and spending more than $800 million. So that's why that most of the pharmaceutical companies, research institutes, national as well as international research institute, and particularly pharma companies, what they have done, or they have started using this computer-aided drug designing approaches. So what is this computer-aided drug design approach, that we are going to see this and how it would be helpful to understand this antibiotic resistance issue, which is a major public health threat right now. So as we know, today we are living in the age of kind of a COVID-19 pandemic, although it is going to end, but still now we are facing some issues, facing some problems. And because of that, we could not go to lab, we could not go to department, universities, colleges, isn't it? But it's being said that after 5 years, so there would be similar kind of situation, because we won't be having a single drug molecule or single antibiotic to kill or to control growth of some of the pathogenic microorganisms. It could be bacteria, it could be fungi. So this is the right time to understand the particular issue, problem, and right now, so give some kind of a solution to this particular issue. So that's why pharmaceutical companies have started the computer-aided drug designing. So one of the applications of like kind of a computational biology techniques or bioinformatics, that one can say, and the specifically technique called as a virtual screening technique. So what is this virtual screening technique? Let us say there is a drug bank or database of drug molecules having a drug-like properties. So those drug-like properties, molecules having in this drug bank, they are sitting, let us, for example, idle. They have not tested for against any specific disease, whether they are active or they are inactive against specific disease, but they possess some drug-like properties. So let us say there are 10 lakh drug molecules are available, and out of this 10 lakh drug molecules, we are going to screen them against only one receptor, one enzyme or one protein. So imagine how much time it would take if we screen 10 lakh drug molecules against one receptor, or for that one receptor, it would take more time, isn't it? Money, electricity, chemicals, everything. It would require in a huge amount. So, but using this simple technique, one can screen all the 10 lakh drug molecules, and one can initially come up to, let us say, 100, then one can come up to 10 molecules and then 1. So likewise, one receptor, one drug molecule. So that way one can try to find out exact location, exact position, the exact amino acids from that receptor, protein, or that enzyme, which can be inhibited or which can interact with the drug molecule or the lead compound. So once we identify that drug molecule, then one can go to library, or one can go to the laboratory and perform some simple experiments like maybe preclinical studies and then enzyme inhibition studies, and then one can go for clinical studies. And this whole process would require nearly 5 to 6 years of time. And after spending less time, cost of that drug would also go down. And the common users like us, it would be easier for us to buy or purchase those drug molecules. That means these drug molecules or these medicines would be affordable to common people like us. So this is the advantage of once we use, particularly that pharma company uses these computer-aided drug designing methods. So this is what today we are going to talk about this. What are these methods and how new drug molecules, new lead molecules can be discovered, can be screened, and can be used. And against how proteomics techniques would be useful in order to understand the target molecule or that receptor molecule or that basically a protein molecule. And then one can perform this molecular docking techniques in order to understand how this drug molecule interacts with the particular receptor molecule. This is what today we are going to talk. All right, so this next slide is just for a student purpose. Actually, just I would like to give some background of what is this particular method and what is this particular subject. So this bioinformatics applications is in drug discovery. So one can predict three-dimensional structures of our interested proteins, enzymes, or receptor molecules. One can predict function of our newly discovered proteins or enzyme, or one can say receptor as well. One can predict gene function, establishment of gene libraries using these bioinformatics methods or some of the tools. Then the ability to use the genome sequences to identify proteins and their function, protein-protein interactions, modifications, and their functions can also be done with the help of these bioinformatics methods. And then one can elucidate the function of a molecule based on its structure. That means once we know the structure, one can predict the function. And if we do not know the structure, one can predict the function or determine the structure using this bioinformatics method. And then one can try to find out the function of the molecule. All right. So the molecular modeling and molecular dynamics simulations, these are the very important techniques right now. Most of the pharma companies are being used widely in the pharma companies in order to predict three-dimensional structure or understand the dynamic behavior of the biomolecules if it comes in contact with the drug molecule. Then next one, conformational search by using quantum chemical methods as well as molecular mechanical methods can also be helpful in order to understand whether the given drug molecule is active or inactive, or what is the most stable structure, most stable conformation of the particular drug molecule before going to interact with that particular receptor molecule. Then molecular docking to understand protein ligand interactions or drug receptor interactions is another important technique widely used by pharmaceutical companies. The data obtained from functional genomics and proteomics could be useful or could be used in the drug designing and discovery process. So at the end, and in the nutshell, this is the particular application of this bioinformatics method to design a drug molecule by using the data obtained by either genomics techniques or proteomics technique. But our final goal is to design a new lead molecule, design a new drug molecule. All right. So, for example, let us say we have a receptor and that receptor is having a nature of as an enzyme or as a protein. Then how does this primary structure of protein or enzyme looks like? So here one can see the FASTA file format that most of the bioinformatics students that would know that what is this sequence. So this is a sequence which gives us most of the amino acid residues. Like for example, first one, we have 9. And second, K for lysine, F for phenylalanine, V for valine, likewise. So each and every amino acid linked in a proper way by holding a peptide bond. And the first one residue is called as a amino terminal residue. And last, like lysine or K is called as a carboxy terminal residue. That means N terminal end and C terminal end. These are two ends are present in any protein, any enzyme, and if that enzyme or protein works as a receptor for that particular drug molecule. So it is very much essential to download the sequence of this particular protein or receptor, which three-dimensional structure we do not know, right? And using homology modeling technique, one can predict three-dimensional structure of this particular protein or enzyme the way X-ray crystallography as well as NMR technique do for that particular molecule. Okay, so such structures are being stored into the primary protein sequence databases, and then those sequences can be analyzed and the results we can put into the secondary databases. I will not go into the details about the product, what are primary databases, what are secondary databases, but there is a technique called as the molecular docking, which using the AutoDock, it is a software tool which is used an automated docking software for the predicting optimal protein ligand interactions. So protein means our receptor, ligand means our drug molecule. That interactions one can try to find out using this particular software called AutoDock. Then molecular docking studies are used to determine the interactions of two molecules and to find the best orientation, best location, best conformation of that ligand, which would form a complex with overall minimum energy. All right, so let me give some specific example about this particular thing, how the system this works about the molecular docking. Just there is one small video here. Let us say, for example, this is our one receptor and this is our drug in the form of protein. So this receptor has got, let us say, active site pocket having a complementary to the active site pocket of this receptor or protein. Then this protein can come in contact with each other, both the proteins, and then there is a formation of a complex called as a molecular docking complex. Similarly, there is another small drug molecule as a ligand, and then we have a receptor or a protein or any enzyme. And if we allow that drug molecule to interact with this protein or enzyme, one cannot get, let us say, proper conformation or proper binding energy. So we try in another way. Another way means we again increase our number of steps to optimize or minimize the particular molecule, and then we get an optimized structure or a stable complex, which having stable molecular energy or molecular geometry of that particular complex, having a receptor and drug molecule. So this is the structure, this is the conformation that we are looking for, and this which can be obtained by this technique called as a molecular docking technique. So this is a very useful technique. That means after using virtual screening, if we screen small drug molecule, so that, and if we have a three-dimensional structure of a receptor, then drug and receptor molecule, one can have a complex using this particular technique. And then we say that, yes, this is the particular drug molecule can interact specifically to this particular receptor. That means once this drug molecule interacts properly with the receptor, there won't be having any kind of a side effects to that particular drug once it's being administered to that particular individual. All right. So this is a very useful technique in order to find out molecular interactions or conformation, suitable conformation, optimized energies for the particular ligand and drug receptor molecules. All right. So that's why the computer-aided drug design or CADD is a specialized discipline that uses computational methods to simulate drug receptor interactions. And CADD methods are heavily dependent on the bioinformatics tools, applications, and databases. As such, there is a considerable overlap in CADD research and bioinformatics. So both the techniques, both the areas are going hand by hand in order to understand proper interaction between receptor and drug molecule. And here is the virtual screening technique for drug discovery. So computational methodologies have become a crucial component in many drug discovery programs, from hit identification to the lead optimization. So lead is a drug molecule. Nowadays, virtual screening has become a valuable in silico technique to search new active compounds, new drug molecules, or new ligand molecules. It is a computational drug discovery approach which is quite an effective screening method as compared to the conventional experimental high throughput screening method. Because if you go by conventional experimental high throughput techniques, it would take a lot of time, it would take years, and again, hard work, extra burden, your energies and chemicals, costly chemicals that would go waste just to screen a single compound from the, let us say, more than 10 lakh drug molecules. But instead, if you sit in front of computers, use some of the techniques, tools, and then one can, out of that 10 lakh drug molecules, one can specifically select a specific drug molecule which could interact properly with the receptor molecule. This is the advantage of this particular method. So the structure, there are two approaches. Like if we know the structure, three-dimensional structure of the receptor or enzyme, one can design a drug molecule. Or if you do not know 3D structure of the protein, based on the drug candidate itself, one can also try to find out which drug molecule interacts specifically with that receptor. This is a major application of this particular technique. Now, after having this background or some introduction about the subject, this is just for students. Just let us come to the main point, that is, what is this antibiotic resistance and drug discovery, and how the proteomics approach useful in this particular technique or in this particular issue or problem to understand. So antibiotic resistance among various infectious agents towards different types of antimicrobial drugs has emerged as a global cause of public health threat. So antibiotic resistance is the ability of an microorganism to withstand the effects of an antibiotic. The diverse newly developed resistance pattern among microbial species and decrease in efficacy of treating common infectious diseases resulted into the development of high levels of multi-drug resistant strains. So this is the major cause, development of multi-drug resistant strains, and how to control their growth or how to kill them in order to get rid from particular diseases. That is the major challenge, that is the major target right now. So antibiotic resistance of aminoglycoside group of antibiotics. Now, what are the aminoglycoside group of antibiotics? Like streptomycin, penicillin, gentamicin, kanamycin. These are the group of antibiotics comes under this aminoglycoside group, which occurs by aminoglycoside kinases. These are the enzymes. Thus, targeting AKs, that is aminoglycoside kinases, from different multidrug resistance could result into the inhibition of AK enzyme and ultimately the resistance. But what is the problem actually? These AK enzymes or aminoglycoside kinases are also being expressed in humans as well, right? As well as some bacterial system they can also express. So if you give one specific antibiotic, so that antibiotic, instead of killing or after killing this MDR or controlling growth of this multidrug resistant organism, they can also try to block the human AKs, that is aminoglycoside kinases. So in order to avoid such cross connectivity, instead of multidrug resistant AKs, they can also target our human AKs, that is aminoglycoside kinases. So designing of the drug is very, very essential in order to avoid certain side effects which may arise after or which can arise after giving the doses or medicine in the form of antibiotics. So here is these techniques becomes very, very essential in order to design a specific drug molecule. So there are 4 mechanisms of this antibiotic resistance, particularly let's say for A, right here. So alteration of the target site of the antibiotics, particularly polymyxin antibiotics. So these are related to several plant materials or plants. They can be targeted this particular polymyxin antibiotic resistance. So what happens? These antibiotics or these bacterial systems, they can change the target site, right? Initially, target site would be like this. So this antibiotic would go and specifically inhibit or block this particular bacteria, but that bacteria can change its target site. Right? So the drug molecule or antibiotics cannot fit here. Similarly, in case of B, the bacterial system can express some enzyme or they can synthesize some enzymes as soon as they come in contact with any specific antibiotic. So that is called as the enzymatic inactivation. So those enzymes can inactivate that antibiotic, or they can change the structure of that antibiotic so that that antibiotic cannot be effective against that particular multidrug resistant strain or multidrug resistant organism. So we work in this particular area, that enzyme inactivation. And then C, here we have active transports of this particular cell. They can try to throw this particular antibiotic through the efflux pump directly without causing any harmful effect to the bacterial system. And then 4th one, that is decrease the permeability of the cell. Cell cannot enter these particular antibiotics inside the bacterial system, so they can just go through the membrane itself. So these are the 4 ways that antibiotic resistance can be, or mechanisms of antibiotic resistance. So we will talk about this one, enzyme inactivation. And these are the 3 enzymes. They are involved in the modification of aminoglycoside group of antibiotics, like aminoglycoside phosphotransferase that we will be talking today on this particular enzyme, that is APH enzymes, catalyzes the ATP dependent phosphorylation of specific aminoglycoside group or hydroxyl groups. These enzymes transfer one of the phosphate group present on the substrate. It could be ATP or it could be GTP to that particular antibiotic, and so that antibiotics becomes a change in structure. There is a change in structure of antibiotics, change in conformation, or there could be two parts that one can get from that antibiotic, so that antibiotic cannot be effective or against that particular specific bacteria. There's the next one, aminoglycoside acetyltransferase. They transfer acetyl groups, and aminoglycoside nucleotide transferases to transfer ANTs. So we will talk today on aminoglycoside phosphotransferases. And these are the group of antibiotics, aminoglycoside group of antibiotics, which we have targeted or which we have studied, particularly gentamicin, kanamycin, amikacin, tobramycin, streptomycin, and neomycin. These are the structures of antibiotics. So today we'll be talking on the specific result that we got on the streptomycin, how the streptomycin resistance can be developed by the particular bacterial system. So therefore, the present study aims to target one of the AKs, that is APH3-1A from the Acinetobacter baumannii through structure-based virtual screening and the test binding affinity of the most stable virtually screened inhibitor with AKs from the Mycobacterium tuberculosis, Acinetobacter baumannii, Enterococcus gallinareum, and E. coli. All right, so this is these results that we had published way back in 2017 in journal of one of the prestigious journal called Journal of Cellular Biochemistry, having impact 4.4. So the molecular docking molecular docking and molecular dynamic simulations that we had performed in this particular research paper, and we had discovered a new drug molecule called zinc 71575419 obtained from the drug bank with the APH from the Mycobacterium tuberculosis, and studied against the Acinetobacter baumannii, E. coli, and as well as into. Though multiple sequence alignment and structure superposition studies have been done, and finally molecular dynamic simulations to understand the dynamic behavior of the enzyme with respect to that particular drug molecule that were studied, and virtually screen inhibitor, how it can interact with the particular receptor. And then binding free energies have also been calculated in order to understand specific contribution given by each and every amino acid towards the interacting to that particular drug molecule. So this is a multiple sequence alignment. Hello, I think someone is talking in between. Please do not talk in between. Okay, so this is a multiple sequence alignment. Then the three-dimensional structures of enzymes taken from different bacterial systems like Mycobacterium tuberculosis, E. gallinatum, E. coli, and baumannii. And we had performed this multiple sequence alignment. And this multiple sequence alignment can be represented the amino acid residues which are nearly common present in between all these three amino acids. And this is the three-dimensional structure of this enzyme called as the APH from this Acinetobacter baumannii was used for the virtual screening throughout the dog cluster. And the NTP binding site, also known as the drug binding site of the AKs and EPKs, and can be identified from this particular molecule. Okay, so these are the drug molecules which we have investigated or identified using the virtual screening technique, and the drug which is having this number, which showed more potent activity as compared to the drugs already available in the market. Okay, so this particular drug called tirfostin, which is available in a drug market right now in the market, and the drug which we have discovered called zinc 71575479. So this drug showed more activity against the already known drug molecule in order to block the activity of this APH or aminoglycoside phosphotransperases. So then what we had done, we have purchased this particular drug molecule and we have purified the enzyme from the isolated strain of the bacillus species. We have isolation, purification, characterization of the drug molecule that we have done, and the drug molecule we have purchased from the market, and we have studied enzyme inhibition studies in particular laboratory, and we got a better inhibition of the particular enzyme with as compared to the already known drug molecules in the market. Excuse me. All right. So this is the binding, these are the binding energies of a particular drug molecule along with different proteins or APH from different microorganisms. And these binding energies can be calculated, and one can calculate using these molecular docking studies, and which can give us an idea that which enzyme is suitable for our drug molecule. And this slide shows us the interactions, molecular interactions between that enzyme, that is APH, against the particular substrate, inhibitor, or drug molecule that we can use. And all the residue that one can see here, like leucine 266, arginine 79, right? And then valine 113. These are the particular amino acids can be called as the active site of the particular enzyme, which can interact specifically with the drug molecule. And that gives us a specific binding of a particular drug molecule with that enzyme. So these are the closed loop having with hydrogen bonding interactions between drug molecule and particular enzyme. But so this slide shows us the dynamic behavior of the complex, that means receptor and drug molecule. Okay, and so this is a control molecule, like magenta color is the control molecule, and as compared to the control molecule, the drug which we have identified shows more stable as compared to the already known stable. So this is the technique, or this is the method called as the molecular dynamic simulations, which gives us idea or information about the how molecules or the drug molecule interact with the receptor with respect to change in time. That is called as the molecular dynamic simulation. This is basically a technique discovered by Professor Martin Karplus, and based on the Newtonian equations called as a particular Newton's second law, that is force equal to mass into acceleration. That we can calculate forces present on each and every atoms present in the protein or in that particular drug molecule, if we know the masses as well as acceleration or supplied by initial acceleration. That is called as F is equal to ma, force can be calculated when with respect to time. That means as soon as there is a change in time, at that particular time, one can calculate all the forces present on the particular molecule, and which gives us a dynamic trajectory or kind of a movie through which one can see that how that drug molecule interacts with the particular enzyme. All right, so this slide shows how the energetic contribution by each and every amino acid residue from that particular enzyme toward that particular drug molecule. Similarly, another research paper that we had published in the same journal, Journal of Cellular Biochemistry, way back in 2018. All right, and in this particular paper also we had done this particular method like molecular characterization of homology search, structural modeling, validation and sequence analysis, molecular dynamic simulations, and catalytic cliff analysis, MD simulations, protein ligand complexes, and MMPBC. This is energy calculation binding studies between protein and ligand, and principal component analysis, which is a new factor or new technique through which one can see dynamic behavior of the molecule, and then in vitro studies, that is inhibition of APH, that also we had done in this particular research paper. So this is the homology model that we have developed, that is stereochemical quality of the optimized APH, amino phosphotransferase, from the bacillus cereus, and the superimposed template. Template means it's a known 3D structure, and the target, which is our structure or our model, which we have discovered, and which shows conserved nucleotide triphosphate binding site or NTP binding domain. That here we can highlight this NTP binding domain, where actually streptomycin, gentamicin, kanamycin, they can interact with this particular enzyme, and that's why we get this particular drug resistance. All right, so this is again pairwise sequence alignment between these two molecules, multiple sequence alignment between these all the four these targets. So I will not talk more on this one, and molecular dynamic simulations the way we had done it for earlier part. So these are the secondary structure analysis that one can try to find out alpha helices, beta sheets present in the particular model. Okay, so this slide shows us the differences between NTP binding domain between all three APH from different organisms like Enterococcus gallinarum, Mycobacterium tuberculosis, then E. coli, and then APH from Acinetobacter baumannii. That how this particular antibiotic interacts with the particular different APH from different organisms. Although the NTP binding site pocket is same, but there are certain differences in amino acid residues, so which could inhibit or which could disrupt the proper binding of the streptomycin of this particular antibiotic to the APH enzyme. So this slide shows us the GTP as one of the important substrate which can interact with the few residues or few amino acids present from the receptor, that is the APH. So here aspartate 195U, which can interact with this particular GTP, that is one of the substrate, and gamma phosphate from the GTP can interact with the two aspartates like 215 and aspartate 195U, and the in presence of these two magnesium ions are here, Mg2+. Okay, the interacting residues are also shown here, and this is the particular molecular interactions of known APH inhibitors containing flavonoids are virtually screened here. So this is another NTP binding site confirmations which we have obtained in comparison or along with the model and then GTP. So earlier we were studied only this GTP, only enzymes, and now this is the enzyme with this particular substrate, that is the GTP against this particular three organisms. All right, so again, these are the stability behavior or stable, how the structure or how the complex is stable with respect to long range molecular dynamic simulations. This is a very complicated and sophisticated technique to study the dynamic behavior of the molecule, whether a complex is stable or unstable, or whether drug molecule is stable or unstable. That gives us the dynamic behavior of the particular molecule. And then here we have calculated binding contribution per residues, that how the each and every amino acid from the active site pocket of that enzyme can liberate certain amount of energy in order to bind with the particular drug molecule or the particular ligand molecule, and that binding energy can be obtained by using MMPBSA method. And this slide shows about the PCA plot, that is principal component analysis. That is this first one, this is without drug molecule, and this is after binding with the drug molecule. That one can see the conformational changes, conformational dynamics can be observed. That means our drug molecule has properly interacted with the particular enzyme, this slide, and this is the result or interpretation of this PCA analysis. Then here cross correlation matrices, some of the hardcore methods are that. I will not talk about this one. let us keep that part. And then another part that which we had done, there's a studies published in Brazilian Journal of Microbiology. So here we had done antibiotic resistance and inhibition mechanism of novel aminoglycoside phosphotransferase, that is APH5, from Bacillus subtilis strain RK. So this is the strain that we have identified as a new strain. We have identified its 16S RNA gene and deposited into the gen bank, and this bacterial system can express one specific enzyme if we are allowed to grow them in presence of streptomycin. And that enzyme is nothing but aminoglycoside phosphotransferase and APH5. So for the viewers or participants here, I would like to inform or mention here that so this is the first time that we have discovered that this aminoglycoside phosphotransferase or APH5 from the Bacillus subtilis strain RK, which is responsible to cause antibiotic resistance against the streptomycin antibiotic. So this is the first report, and we are glad to inform you that this is a new type of enzyme we have discovered, right, from which is responsible to cause antibiotic resistance. So this is using some of the proteomics approach. So after purification, isolation, characterization, and purification of that particular enzyme, we have identified its complex interactions with, let us say, for example, using HPLC, then LCMS, right? And then we have discovered or deduced this model, or not model, but actual mechanism that how streptomycin can be inhibited or growth of the particular bacteria can be inhibited by this particular enzyme. And the drug which we have identified shows more inhibition as compared to the streptomycin. So this slide shows us the purification part actually, and these are the markers that we have used. And our enzyme, which is expressed, that is APH5, is more expressed in from the Bacillus cereus with 34 kDa dozen approximately. That means as soon as we grow this particular bacteria, that Bacillus strain, RK strain, in presence of antibiotics, then this is the particular enzyme we found as a more expressed or more produced by that particular bacteria. Why? Because this is the enzyme can block the activity of the streptomycin, and that is called as the antibiotic resistance. So this in vitro studies of APH from Bacillus cereus, ATCC, and a couple of spectrophotometric assays, free lysis shows us that drug molecule can inhibit the property of the particular antibiotics. All right. Then another part of the study which we had done, that is published in Journal of Ethnopharmacology, one of the prestigious journal, that biofilm inhibition mechanism from the extract of Hymenocallis litoralis leaves. So this is the basically collaborative research work between Department of Microbiology and Department of Biochemistry, along with animal studies that we have done in this particular studies. So what we have done, this is a particular plant extract plant, which is ornamental plant basically present in our university campus. So we had taken leaves of the particular plant and extracted certain plant extracts or phytochemicals from this plant, which showed us which presents alkaloids, flavonoids, then certain potassium hydroxide flavonoids, terpenes, terpenoids, then glycosidases, phenolics. These were the compounds present in the particular plant extract, that is leaves extract. And we had tested those particular phytochemicals against certain pathogenic organisms like Candida albicans, Staphylococcus aureus, Bacillus subtilis, Salmonella typhimurium, and Proteus vulgaris as a part of methanolic extract, and which showed inhibition in terms of millimeter. So this is the growth curve pattern of Staphylococcus aureus NCAM 2654 strain in the presence and without plant extract. So this part shows us that in presence, in absence of plant extract, there is a proper growth of this pathogenic bacteria called Staphylococcus aureus. But after use of this particular plant extract, there is an inhibition that we have found. That is, there is a growth inhibition in this about this particular extract. Similarly, about this Candida albicans also shows this is a particular growth in absence of plant extract and in presence of the plant extract. That means these phytochemicals are very much useful against this particular pathogenic microorganisms like Candida albicans as well as Staph aureus. These are the deadly pathogenic microorganisms. So this SEM image shows us that this is the particular arrows. It shows us that the particular growth can be inhibited by the particular plant extract and phytochemical present in the plant extract. And these are the without plant extract. That means here we can biofilm formation can be seen. The microorganism growth is properly seen. But here in presence of plant extract, there is no growth or inhibition. Yes or yes. Similarly, in case of Candida albicans also growth is inhibited, or in absence the growth can be in a proper way. right? So this can be inhibited by the extraction by the supplementary actions given by the plant extract. And then what we had done, these are the phytochemicals that we have identified by LCMS technique. Initially GCMS and then LCMS confirmed by LCMS technique. And all the components are present in our extract. And that extract that we had tested against the histology study to see the rat skin after 24 hours treatment. And the figure 1A is a normal or control that we have taken. And after administration of this 100 μg, 500 μg or 1000 μg of that particular microgram methanoic extract, and there is no any harmful effect that we could see on the particular rat skin. That means the plant extract which we have obtained or phytochemicals which are present in the plant extract, they cannot cause or they are not that much of harmful to the animal skin. That means these drug molecules can be used suitably in order to kill the pathogenic growth of the Candida albicans as well as Staph aureus. And these are the certain drug molecules which we have identified by using LCMS technique. So this each drug can be studied independently as an independent drug molecule against those particular things. Now, what is this proteomic approach in this particular thing? So what we have done, we have taken two enzymes like sortase A from the S. aureus, right? And the drug molecule which we have identified by using LCMS technique, those drug molecules that we have taken it here and studied molecular docking studies to see that why these particular phytochemicals have inhibited the growth of these particular pathogenic microorganisms, right? Which proteins are involved, which enzymes are involved. So this sortase A from the S. aureus was responsible to interact with this particular enzyme, and one can try to block the particular enzyme by these phytochemicals from this Hymenocolic plant extract. Similarly, this protein called ALSL3 from the C. albicans, that is Candida albicans, is responsible to form anti-biofilm, to form biofilm. And because once the organism forms biofilm, then one cannot whatever drug molecule that we use in order to cut down this biofilm, they are not that much useful. This plant extract could be useful to block the activity of this ALSL3 from this Candida albicans. Now, currently the chemical antibiotics are used as a sole therapeutic components to control various infectious diseases. But many pathogens have developed antibiotic resistance against these therapeutic agents. So here one can see the pathogenic microorganisms here with the shield, right? And needles are here. And doctors with molded needle, they are a little bit confused that how to control or kill those particular pathogenic organisms. So that's why it's being said that antibiotic resistance is a big threat to the society. So it is necessary to discover new therapeutic agents to fight against these pathogenic entities. So what would be the solution? The solution is nothing but a small peptide called as antimicrobial peptides. Again, proteins or small peptides. So these small antimicrobial peptides can be extracted, unified, and characterized using the proteomics approaches or proteomics methods. And those small AMPs can be used against some deadly pathogenic microorganisms like Pseudomonas aeruginosa, Staphylococcus aureus. So here we have used those AMPs in presence of antibiotics, right? As an independent antibiotics, we could see some zone of inhibition, and AMP, we can see some zone of inhibition. But if we combine both of them, antibiotics with the neomycin, with the AMP, that is antimicrobial peptide, one can see bigger zone of inhibition. That means this AMP and antibiotics are more useful in order to control growth of this Pseudomonas aeruginosa. Similar in the case of Staphylococcus aureus. All right, here we have used kanamycin. Again, the zone of inhibition is more that one can see here, right? And then a bacterial system that we have changed, like Bacillus subtilis, Bacillus cereus, Staphylococcus aureus. So this AMPs, that is small antimicrobial proteins, but antimicrobial peptides having less than 20 amino acids are very, very useful if we use them in presence of or along with antibiotics in order to control growth of the pathogenic microorganisms or kill them. So this was about the, let us say, for example, role of this particular antibiotics with respect to proteomics approach. But if you have to block this particular, because most of these antibiotics like streptomycin, gentamicin, penicillin, basically streptomycin, they are protein synthesis inhibitors. So in order to identify how these antibiotics can inhibit the protein synthesis process, then one has to go at this protein synthesis process itself. And the protein synthesis process is nothing but controlled by the important molecule called as a transfer RNA. And this transfer RNA contains different modified nucleosides present at the specific positions 34th and 37th. And these are the three anticodons like 34, 35, and 36. And these are the modified nucleosides present in that particular positions. Like uracil can be modified to MNM5S2, or adenosine can be modified to M6A, guanosine can be modified to M1G. These are the modified bases that one can try to see. And if you see the three domains of life like bacteria, eukaryote, and archaea, most of the time modified nucleosides can be observed. So these modified nucleosides, these are the some of the structures or chemical structure, molecular structures of these particular modified nucleosides. Like some of the modifications at the ribose base, ribose ring. Some modifications are at the base position. At simple modification like CH3 groups can be added. But there are certain modifications which can be added as a complex chemical molecule, like addition of single amino acid itself. So that's why this cytosine can be converted into a lysine, that is by the addition of lysine amino acid at the particular C2 position. So these are called as larger modifications. And these modifications are done by certain enzymes secreted by the particular species. And those species can secrete or do these particular modifications. Okay, so this some molecular dynamic simulations and change dynamic behavior of the molecule in presence and absence of this modification that we had done, we had studied, and we know that this particular molecule, this is a particular research work that we had done, published this paper in Cell Biochemistry and Biophysics on this particular modified nucleic acids. So I will not go into the detail of this particular studies. Just I will at the end, I would just like to see the research contribution given by this our department, that we could we had given one new model called as a footstep model, which shows two hydrogen bonding as well as a single hydrogen bond, and which has resulted as a one of the extension to this the Wobble hypothesis, which was put forth by Francis Crick in 1966. And this Crick is one of the author who had shared Nobel Prize in 1962, prize for the discovery of double helical structure of DNA, that how one tRNA comes in protein biosynthesis process and how it can leave the process. That was the thing, but they did not say that time how is the entry takes place and how they can leave the process. So that study that we had done, and based on that we had given one different model or new model called as which studies had been published in the prestigious journal called PLOS-1. And then similarly, these antibiotics and ribosomal systems, tRNA, mRNA complexes can be studied. And here a double hydrogen bond, single hydrogen bond we identified. And this model that we had given name as a footstep model, which can recognize the proper codons during the protein biosynthesis process. And this is the one of the extension to the Wobble hypothesis put forth by Francis Crick, who had shared Nobel Prize in 1962. So this is one of the good contributions from our laboratory, that we had supported this Wobble hypothesis. But not only that, we had given extension of that particular model, and we called as a footstep model. So this study had been published in prestigious journal, and the paper which we had published in Cell Biochemistry and Biophysics way back in 2013 has received very nice recognition, and the paper had been cited by the prestigious journal called Cell 2018, having impact factor 38.66. Then original Nature paper for having 42 impact factor, Nature Communication Biology, then Nature Scientific Reports, and Molecular Therapy Nucleic Acids. Similarly, another research paper which we had published in Cell Biochemistry and Biophysics way back in 2015 also got citations in Nature's Use Microbiology, having 34 impact factors. So at the end, effective drug molecule can be designed using this proteomics approach, particularly bioinformatics and experimental proteomics approaches. And one can understand structure, function, and dynamic behavior of the biomolecules, as well as some cellular processes in detail at the molecule level. So these are the research collaborations. At state level, we have 7 collaborations. At national level, we have 3 collaborations like CSN, Institute of Chemical Biology, Kolkata, Indian Institute of Science, Bangalore, National Institute of Virology, Kerala unit, and international level, there are Aalto University, Finland. We had published 1 research paper in ACS journal, and University of Jahangirnagar University, Dhaka, Bangladesh, 2 research papers have been published, and KwaZulu-Natal University, Durban, work is in progress. So these are the research students that we have contributed in this particular work, particularly Rishikesh, Sagar, as they have done work on this proteomics approaches of antibody resistance. Prayagraj, then Asmita, they had done work on modified nucleic acids in order to identify the exact role of modified nucleosides in protein biosynthesis process. So I would like to acknowledge here Department of Science and Technology, New Delhi, University of University Grants Commission for providing funding, Lady Tata Memorial Trust, Mumbai, for providing fellowship to the students, and heads of the departments of biochemistry, biotechnology for providing infrastructure facilities. And I would also like to thank our Honorable Vice Chancellor Professor D.T. Shirke sir, and Honorable Pro Vice Chancellor Professor P.S. Patil sir for allowing us to use university infrastructure facilities. And similarly, Honorable former Vice Chancellors Professor Devanand Sindhe sir, Professor Dr. A.M. Salanke sir, and Professor Dr. N.J. Pawar sir for allowing us to use the computational power available at Computer Center's team, Shivaji University, Kolhapur. So thank you very much, one and all, for your kind attention, and I will be very much happy to have some questions. Hello, sir, over to you.
So,
हॅलो हा पटवारी सर
yes, yes. आवाज येतो ना?
हा येतो सर
Topic is open for discussion. If you are having any questions, query, please ask. From audience, from participants, if you are any questions, please ask.
सर actually I want to ask some questions.
Yes, sir.
What is the role of protein in metabolism?
What is the role of protein in
Metabolism.
metabolics? Yeah. So, for example, let us say antibiotics is our drug molecule. So, as soon as we give any antibiotics to that in order to control the growth of that microorganism, it could be bacteria or fungi. So that antibiotics or that bacteria can express some proteins, right? Highly expressed proteins, and those enzymes as a metabolic rate. So those enzymes can try to change the conformation of that antibiotics. And that's why the antibiotics is not that much effective the way doctors think. Because of that enzyme is expressed by that bacteria, if it comes in contact with the antibiotics, if it is streptomycin, gentamicin, kanamycin, and because of this particular complex, that antibiotics becomes inactive. So that is the role of this particular metabolics. All right.
Any questions from audience, from participants? Yes.
Sir, vote of thanks, Mama.
No questions?
No.
If no questions,
All right.
then I can conclude the session. I am very much thankful to Professor Dr. K.D. Sonone sir, which has given a beautiful, nice lecture, nice talk on bioinformatics applications in drug discovery. In his talk, sir covered the aspect of computer-aided drug designing and molecular docking techniques. Also focused on antibiotics resistance and drug discovery, antibiotic resistance and its mechanisms. We enjoyed the whole lecture, very nice lecture it was. Also, sir, on antimicrobial activities of purified AMPs. I'm very proud to say, sir has published his research papers in the well-reputed journal Nature, which has impact factor 38.66 and 48.778. It is beyond imagination. Actually, the Nature journal, we know, sir has its own contribution, his research group has own contribution in this journal. Personally, I congratulate Dr. Sonone sir, and thank you very much for giving here a beautiful, nice lecture. We enjoyed, sir. I am very much thankful to convener of this conference, principal, management members, and all dignitaries from the organizing committee who gave me a chance as a chairperson to this session. Thank you very much. With the permission, I stop here to conclude this session. Thank you very much.
Thank you, Professor S.B. Patwari, sir, for your valuable discussion and handling session second. Also, my sincere thanks goes to Professor K.D. Sonone, sir, for wonderful presentation. Now, we will start the session third. Once again, I welcome all in session third. The session coordinator is Mr. R.S. Gaikwad, sir. I request Mr. R.S. Gaikwad, sir, to handle the session. Gaikwad, sir.
Okay, thank you very much, Gombre sir. Now I am starting session third. As a chairperson, Dr. Gaikwad Shashikant Harishchandra, having the qualification MSc Set PhD, very dynamic personality. Now he is working as a professor Shri Shivaji Mahavidyalaya Barshi and also working as a chairman BOS in chemistry Punyashlo Kahilya Devi Holkar, Solapur University, Solapur. Teaching experience is near about 28 years. Research paper published in national international level is near about 27. Research paper presented in national international conference is about 33. Research guided student awarded 3 students and now enrolled 3 students. Then UGC MRP completed 3 projects. Invited lecture and chair session, chief guest at national international conference is about 12. Good scholar citation, so research gets score 15.26 or cheat. Is also Scopus citations about 140. Life member of ASSET, BRC, India, NUCAR, BRC, ISC, ACS, USA. Training completed is near about 4. Coordinator, IYC, SAMQs, 2012, ALS, GLP, 2013, state level workshop. Equal opportunity cell and special cell, NetSet workshop. College coordinator, Solapur University, Aviskar Research Festival. Observer for the set examination conducted by Pune University, Pune. Organizing committee members for Solapur University Youth Festival, International Conference of Solapur University. Awards achieved. The reviewer of elsewhere, Taylor Francis, Springer, JCSP, OJS, RJC, Material Focus, FIP Fellowship under 9th Plan of UGC appreciation by Lupin India Limited, Thane, for supporting their Earn and Learn programs. Appreciation by Lions Club Barshi, SCST Employee Federation, Solapur, SSPM Barshi. University level committee also selection of principals and teachers, BOS, then other some committees, then RRC, DRC. Such a long profile of Professor Gaikwad Shashikant. Now I request to Professor Shashikant Gaikwad to continue their further session.
Thank you, dear friend, respected Rajkumar Gaikwad, sir, the head of the department, Vidyan Mahavidyalaya Sangola. Thank you for a kind introduction. And friends, today we are here in this national conference, which is conducted by Vidyan Mahavidyalaya Sangola through the online mode, that is on the Zoom platform. And I welcome you all in the third session. And in this third session, before going to this third session, I would like to pay homage to the founder of Vidyan Mahavidyalaya Sangola Institute, respected Ganpatrauji Deshmukh sir, who played a very important role in the politics of Maharashtra. And he is the pioneer for such a great institutes at a rural level in Sangola. So it's my prime duty to express gratitude towards respected Ganpatauji Deshmukh sir. Now I shift over to the main function, the third session, and for this today's third session, the chief guest is Dr. Balaji Rokde, and I will introduce Dr. Balaji Rokde. So this is me a great privilege to take a chair as a chairman in which the renowned scientist Dr. Balaji Rokde will deliver a lecture in this conference. So here is the brief account of Dr. Balaji Rokde sir. He is a PhD in organic chemistry from Department of Organic Chemistry, the prestigious Indian Institute of Science, Bangalore, and he's completed the PhD in the July 2014. He's a master in organic chemistry from 2006 to 2008 from the Sri Ramananda Tirth Marathwada University, Nanded. He also completed BSc from the same university in 2003 to 2006. After completion of BS MS from Nanded and completing the PhD from the ISC Bangalore, he's continued for his scientific career, and at present he is a process chemist and scientist at Eli Lilly Kinsale Limited from April 2021 from Ireland. He is a postdoctoral fellow at School of Chemistry, University College, Dublin, Ireland. During the October 2015 to the March 2021 is a long period as a postdoctoral fellow at Dublin. He also worked as a research associate at Department of Organic Chemistry, Indian Institute of Science since 2014 to the September 2015 at ISC Bangalore, India. He also worked as a teaching instructor, especially general chemistry, from 2017 to the November 2017, September 17 to November 17 at School of Chemistry, University College, Dublin. So to his a great experience, he also worked as a teaching assistant, particularly for the organic chemistry, Indian Institute of Science, Bangalore, since August 2012. He is a volunteer teaching in high school science teachers training program, which has been conducted by the Indian Institute of Science, Bangalore, from 2011 to the September 2014. So from this one we can recognize that he is a fond of teaching as well as scientific temper. So because of his scientific temper and teaching affinity, he has, to his credit, various awards and fellowships. Few of them are listed here. He is a scientific editor in the prestigious Royal Society of Chemistry's Literature Updating Service, Synthetic Reaction Updates, from 2015 January to the October 2020. He is a member of Royal Society of Chemistry RSC Advances Review Panel from January 2015 to this till date. He has a postdoctoral fellowship from Biomedical June 2019 to the March 2021. He also awarded a postdoctoral fellowship for the Synthesis and Solid State Pharmaceutical Center from October 2017 to the May 2019. He also has to his credit international postdoctoral fellowship by Irish Research Council during the period of October 2015 to the September 2017. He also awarded a PhD research fellowship by Council of Scientific and Industrial Research, New Delhi, India, in August 2009 to the July 2014. It means that he has qualified the CSIR's national level test for the research. So national level exam, he has been qualified for the lectureship in 2008. He also qualified the graduate aptitude test GATE examination for engineers in February 2009. And he has also, to his credit, a state eligibility test, that is SET, which is conducted in May 2009. To his credit, as a scientist, he's contributed as a reviewer for the various international journals. He has reviewed up till now 52 research articles, and he has recommended whether to accept or not such a long number of articles. He has to credit chemical communication, 13 research articles, RSA Advances, 38 research articles, and Monsef for Chemi, 1 research article he has reviewed up till now. And we will have a great interaction, such a renowned persons, where to his credit we found that 14 research publications, 1 book chapter, and as a scientist, to his credit, at this stage he has 518 citations. So he has up till now presented various articles and presented research papers at international national conferences. So this is a great achievement to the credit of Dr. Rokre sir, and we have with us a great scientist, Dr. Balaji Rokre. I warmly welcome him to this conference and request Dr. Balaji Rokre sir to present his research in this gathering of students and teachers. I hope this will be definitely helpful to the budding scientists, those are studying at Vigyan Mahavidyalaya Sangola, and researchers, those are around us. Thank you. And again now I welcome Dr. Balaji Rokre sir. Here is Dr. Balaji Rokre. Thank you.
Am I audible?
Yes, sir.
Yes,
Okay.
sir, it's audible.
Yes, sir, definitely.
Thanks. So I would like to thank, first of all, for the organizing committee to invite me for this nice conference, actually. Particularly my friend, Dr. Siddharth Kadam, who is here in the audience. So yeah, we are very close, actually. So one day he called me and he said, yeah, there is a conference. Would you be coming? I said, yeah, why not? So yeah, I know the name of the conference is, the conference is about proteomics, but I'm an organic chemist, so I'll talk a little bit about organic chemistry. I hope there are chemists in the audience who will understand what I'm talking. So with all this, again, I would like to thank Gaikwad sir for warm or kind introduction. It was quite long, so thank you for that. So with that, without wasting time, I'll start my presentation. So what I'll be doing is I'll be speaking only one work from my postdoctoral research. So yeah, right now I'm a senior scientist in Eli Lilly, which is a pharmaceutical company. You guys might have heard of Pfizer, right? So because it's well known for vaccine, the COVID vaccine. We don't have any vaccine, but we do have the tablet actually for patients who are in the hospital. So yeah, Lilly is one of the well-known pharmaceutical company. So yeah, I'll start with the presentation. So today I'll be talking about the axially chiral PN ligands for asymmetric catalysis. So for the students, okay, yeah. Okay, so I'll talk a little bit about asymmetric catalysis, so because I'll be talking about asymmetric catalysis, a student might not be aware of what is asymmetric catalysis. So you might have learned about chiral compounds in your undergraduates or masters, right? So, and also you might have heard about catalysis. So asymmetric catalysis is nothing but, it is a type of catalysis in which you use chiral chiral catalyst to generate chiral compounds. So generally what you get during this asymmetric catalysis is only one form of the chiral molecule. So you might have learned R and S in your undergraduates or postgraduates, so 2 enantiomers and all. So yeah, that's what you generate by using asymmetric catalysis. So for you can also call enantioselective catalysis because you can selectively synthesize one enantiomer from the 2 possible enantiomer, right? So why this field is important? In 2001, there was a Nobel Prize for asymmetric catalysis. So the chemist who got this Nobel Prize was KB Sharpless. So you might have heard about Sharpless epoxidation. He's the guy. Then Professor Noyes and Roji Noyeri. So those 3 guys got the Nobel Prize for the development of catalytic asymmetric synthesis. And very recently, last year, Professor Benjamin List from Germany and David Macmillan from Princeton University got a Nobel Prize for the development of asymmetric organocatalysis. So this is why the organic asymmetric catalysis is important. So apart from that, the synthesis of enantiomer is very crucial in various fields of chemistry, including pharmaceutical, biological, agricultural, fragrance, and material chemistry. So if you look in the pharmaceutical industry, generally like almost 56% of the drugs are chiral in nature. So it is very, very important, to develop to develop a asymmetric route to synthesize chiral molecules. Okay, yeah. So if you want to do the asymmetric catalysis, you need to have a chiral catalyst. Okay, so you can't make a chiral compound without having any chirality in the reaction mixture. So there has to be something chiral to generate chirality. So if you don't have the chirality. So how you can make chiral catalyst? Okay, so there are various ways. So I'll talk about one of them. So first of all, what you can do is one of the ways to use the chiral ligand. Okay, so you have a chiral ligand, you synthesize chiral ligands. So a lot of researchers have developed various chiral ligands. So you have a chiral ligands, and what you do is you react that chiral ligand with a metal So you have a metal salts, various metal, you take copper, palladium, or any iridium, rhodium. So you can make a chiral complex with the chiral ligand, and that will act as a asymmetric or your chiral catalyst. So what you do is you use that catalyst in chemical reaction, you add it into the reaction, and so what happens is the chiral ligand will transfer its chirality to the reaction product, which is why your product becomes chiral. So you know the catalysis. What happens in catalysis is you use, say, one equivalent of catalysis, which can turn over many more equivalents of your reactant into the product. So you use a very, very small amount of catalyst to generate a number of product molecules, right? So that's why it is called catalysis. You just need a little bit of catalyst to catalyze the reaction. So the ligand controls the binding of reactants and their subsequent reaction paths through the combination of steric and electronic interaction. So more commonly, if you look at the ligands, so you have a donor atom. So donor atom could be phosphorus, it can be nitrogen, it can be oxygen, or it can be sulfur. So if you see any ligand, these will be the the items you will see in the ligands, okay? So, and they will help to electronically tune a metal center. Okay, so these are some of the examples of chiral ligands. So you can see, so this is a ligand called Salen ligand. So herein you can see the chirality over there. So you have two chiral centers. So one is here, other is there. And this is other ligand. So it can be binol if it is X is OH. It can be binap when the X is diphenylphosphine, or if it is NH2, it can be called as a binam. So you can see that there is a chiral axis over here, right? So in the first ligand, you have chiral center. In the second ligand, you have chiral axis. So there are various kind of chiralities. So you have, but we'll stick to only two here. So I'll not confuse you guys. So this is the chiral axis. So again, this is tadol. So you have a central chirality here over there. So this is box ligand. You have a central chirality here. This is Joshi phos. So this is ferrocene based ligand. So here you have a planar chirality. This is third kind of chirality. So here this is the ligand called phosphonimidates. So here again you have a chiral axis. And this is the Fox ligand, one of the very well known ligand. Herein you have a central chirality, and this is the ligand called DPEN, wherein you have two chiral centers. So these are the the chiral. So here the central chirality, chiral axis, and the planar chirality will help in catalysis to generate the chiral products. So okay, next. So I'll talk about my work. So I'll slowly introduce. So as I mentioned, you might have heard in earlier slide about the Professor Noeri. He is the one who developed this binap catalyst. So if you look at the binap, it has a chiral axis over here. So that is why it is chiral, right? So this is one of the well-known axially chiral molecules. So because it has a chiral axis, we can call it axially chiral. Okay. So this is C2 symmetric in nature. So what happens is because of C2 symmetry, it reduces number of possible substrate catalyst conformation, and it also reduces number of potential reaction pathways because of C2 symmetry. Those are the advantages. Sometimes two identical donor atoms, so here you have a phosphorus as a donor atom, interact differently with the metal catalyst during the catalytic cycle. So then the Professor Hayashi, who is from Japan, what he did is he replaced one of the phosphine atom with methoxy. So with the hydroxy, you have two different donor atoms. You have phosphorus and you have oxygen. So that will give completely different chiral catalyst, right? So because you have two different donor atom. So we hear in Professor Brown, who is from UK, Oxford University. He's the postdoc supervisor of my postdoc supervisor. So he's kind of my academic grandpa. So what he did is he replaced, this above naphthalene moiety with this quinazoline moiety, wherein you can see now you have nitrogen, but this is within the ring. So when the metal form complex, so it will be like so in this case, in this and BINAP and MOP case, you have 1, 2, 3, 4, 5, 6. So 6 plus your metal, so you have 7-membered metallocycle, wherein in QNAP case, so you have 1, 2, 3, 4, 5, and 6. So you have metal sitting over here. So you have a 6-member metallocycle, right? So yeah. So how this axially chiral PN ligands have evolved over the years. So the first ligand, which was QNAP, was prepared by Professor Brown in 1993 in Oxford. Later on, Professor Gairy, who is my postdoc supervisor, synthesized this ligand called quinazolinab. So you have this quinazoline moiety over here. That's why it is a quinazolinab in 1999. Later on, Professor Chan came up with this ligand called PIFOS. In 2004, Professor Carrera came up with this ligand called PINAP. So in this PINAP, you see that there is a extra chirality over there. So in other 3 ligands, you have only the chiral axis, but in PINAP, you also have a chiral center over here, right? So similarly, then in 2006, Professor Gairy developed this ligand called quinazox, which also has the chiral axis as well as the chiral center over here. And more recently, Professor Afanik from University of Florida came up with this ligand called STACFOS, which has, so now you see in earlier, all the 6 ligand, you have 6-member nitrogen containing moiety, but here this is 5-member, and it's very difficult to introduce the 5-member moiety. Actually, the reason being is if you have a 5-member moiety, then the chiral axis will not be stabilized. So what happens is it will racemize at room temperature because you don't have this extra phenyl moiety, which will sterically, hinder with a down phenyl moiety, naphthalene moiety, and that's how you stop the steric rotation around your chiral axis. But if you have 5-member ring, you don't have the extra phenyl moiety, and therefore the chiral axis will not be as stable as your other ligands. So what he did is he introduced, so because you have this extra nitrogen over here, so he introduced this pentafluorobenzyl ring, so which is electron deficient, and you have a naphthalene moiety down here. So what happens is they will interact with each other, and because one is electron poor and other is electron rich, so there will be interaction between them, and they'll have a kind of pi stacking, right? Benzene ring has a pi electron, and it will have a pi stacking between electron poor and electron rich rings, and that's what stabilizes the chiral axis. So with that, okay, so I'll talk about disadvantages and advantages of these ligands. Actually, if you see all these 4 ligands over here, so the problem with that, these ligands is, so once you make these ligands, they will be in a state of racemic mixture. So you have both the enantiomers, you have R as well as S enantiomer. Then what you do is once you make that, then you use some resolving agent. So in this case, it is this chiral palladicycle, so right? This is palladicycle. So you use this as a resolving agent. So you react with your racemic mixture of these ligands. So what happens then is this palladicycle will react with this, and it will form a diastereomer. So this is earlier you had a mixture of enantiomers. So, and because you are starting with the chiral palladicycle, it will form a diastereomer. So, and you learned in your bachelor's or master's, right? So diastereomer has completely different properties if you have, say, say you started with this yes Faraday cycle. So you have a yes chirality here. So what happens is you have yes chirality constant here and yes chirality constant here, but your enantiomers of your ligand will react differently, and it will, because now you have a two chiral factors. You have chiral axis and you have chiral centers. So these are the diastereomers. Since diastereomers are different, they will behave as a completely different molecule. You can easily, you can do column chromatography, you can purify them, or you can do a nice crystallization. You need to put some efforts actually to do that. It's not that easy, but compared to isolating enantiomer, it is quite easy. But again, the problem here is you need to use this chiral Faraday cycle, which is quite expensive in nature, and sometimes, as I said, the crystallization of these diastereomer is not that straightforward actually. So but yeah, so these other two ligands which has the other chirality factor inside them, you have a chiral center over here in PineApp, and you have a chiral center over here in Quinazox. So what happens is you don't need the chiral Faraday cycle, because there are two chiral things in your ligand itself. So it can act as a inbuilt resolution, and you can get the diastereomer formation, right? So once you have diastereomer, as I mentioned earlier, you can easily separate these isomer by column chromatography or by crystallization. Again, you need some efforts there, but it's quite easy as compared to using the chiral Faraday cycle and all those sort of things. So it can also, as I mentioned, you can avoid palladium amine complex, which are expensive in nature, and this extra chiral units can be beneficial during the reaction, right? So as you have extra chiral things, your enantiomeric excess of your product could be more. So with that, yeah, okay, so I'll talk a bit about this FOX and FIM. So what is FOX? So FOX is the short form of phosphino-oxazoline. So you have phosphine here, and you have this oxazoline moiety. So these are very well-known ligand. Actually, in asymmetric catalysis, the guy who, or the people who works in chiral chemistry will easily identify this. So these are one of the best PN ligands available in the literature. They are modular at chiral center, modular means you can easily change the R group with various groups, right? You can have phenyl, alkyl, or whatever. So you can change it there, and you can also, because you can make chiral phosphine, right? So if you have three different moieties on the phosphine, you can make phosphorus as a new chiral center. So you can modify your ligand both at this chiral center as well as at the phosphorus moiety. And this ligand has been used in various catalytic reactions successfully. And on the other hand, there is this phosphinoimidazoline. So you have the phosphine group and you have imidazoline ring here instead of oxazoline ring. So that's why these are called phosphinoimidazoline, or short form PHIM. So these are also known in the literature, but they are not as well as explored like oxazoline ligands, because there are various issues like synthesis is a bit tricky, and as I mentioned, they are also modular at these chiral center and the phosphine moiety. And apart, the additional thing is you have an extra nitrogen over here as compared to FOX ligand, which you can fine tune. You can have various group. You can have electron withdrawing group over here. You can have electron donating group. So what that will add is, you can tune the electronic nature of this nitrogen, because this is directly coordinating with your metal. So if you want to make your metal electron reach, you have a electron reach R group over here. So you can make your metal center electron reach. If you want to make it electron deficient, you have an electron withdrawing group as your R group. So you can pull out electrons from the nitrogen and you can make your metal center electron deficient. So it's very, very important actually to tune the properties of your metal. So with that, so what we thought is we took some clue, clue from the Apony group, who has developed this stacked phos ligand. And we thought, okay, what we can do is, so because this required chiral paradicycle to resolve, and we thought, okay, we can combine these two things together and we can make a very new, like, or a novel ligand, which has never been synthesized earlier. So which is this ligand over here. So what's the benefit of this ligand? So you have, again, like central chirality, you have a axial chirality, right? And as I mentioned earlier, the configuration stability means your chiral axis is stabilized by the pi stacking between your electron poor pentafluorobenzyl group or the pentafluorophenyl group and your electron rich naphthalene. So it is modular and tunable at both this chiral center as well as the phosphine. And you can you will have a ligand with modified bite and dihedral angle. So bite angle is what is bite angle? So when your ligand forms a complex with the metal, so you will measure the angle over there and you will see there will be some angle, so which will be like different, compared to all the known chiral ligands. So these are the advantages with this ligand if it is successfully synthesized. So with that idea in mind, we started, we put, the retrosynthesis. Okay. So we thought that, okay, we can install this diphenyl phosphine moiety by nickel catalyzed cross coupling between the diphenyl phosphine and this triplet moiety over here. Then you can generate this triplet moiety from the corresponding methoxy naphthalene, alkoxynaphthalene. The alkoxynaphthalene can be synthesized by the N-benzylation of this imidazoline substrate. This can be easily synthesized from this aldehyde, alkoxynaphthaldehyde, using NBS mediated cyclization. So in the reverse, in the forward sense, we started our synthesis with this chiral moiety over here, which is like 1,2-diphenyl ethylene diamine. So chiral centers are fixed here. So you have a SS chirality over there. So this, oops, okay, what happened? Excuse me, okay. So yeah, you have this SS chiral centers over there. So you react with this 2-methoxy naphthaldehyde by using N-bromosuccinamide in DCM as a solvent. So you generate this imidazoline product in 80% yield. So then once you have this imidazoline moiety, just react with base. So sodium hydride will act as a base. It will take up this proton and you have this benzyl bromide over here. So it will react with this in SN2 manner to generate this benzylated imidazoline in 1:1 diastereomeric ratio. So now you are here seeing the chiral axis, right? So as soon as you add this pentafluorobenzyl moiety, you see the chiral axis in the NMR or in the molecule. So yeah, that can be easily confirmed by NMR. So you can see those different chemical shifts. So this was synthesized in 80%, 80 to 78%. Then we did the demethylation of this methoxy group by using BBR3. And then once you have the phenol over there or the naphthol over there, so you can protect that with triplyc anhydride to synthesize corresponding triplet. So the yield of this triplet was 63%, was 2 steps. So again, the DR ratio was same as corresponding methoxy naphthalene. So then we did final nickel catalyzed cross coupling between diphenyl phosphine and this triplet, which furnished the expected ligand in 49% yield. But now you can see that the ratio of DR was changed from 1:1 to 3:1. Okay, so you get 3 of one enantiomer or one diastereomer and one of other diastereomer. That is because, like the diphenylphosphine is quite bulky, and that's what one of the diastereomer more stable as compared to the other. So then once we have this mixture, we did a fractional crystallization in diethyl ether. Wherein you can easily crystallize out the major isomer, which is like 3 in 99.5% of DE. So the ligand was prepared in 5 straightforward steps in overall 19% yield, which is quite good. There is a lot of work actually behind this single slide, but this is like almost 1 year work, but you can see like only 5 steps here, but it has taken a lot of efforts. So then we established the chiral axis, like the configuration of chiral axis. So we generated the crystals and we sent it for single crystal X-ray. And as you can see that it was found to be R. The chiral axis was R configuration. So then we've also found that in X-ray crystallography that the pi stacking was seen in between this pentafluorophenyl group and the naphthalene moiety. So the distance between this ring and the naphthalene moiety was found to be 3.5 angstrom, and that is what expected to be seen actually if you have the substrate like this. Then what we did is we wanted to study the stability of the chiral axis, like how stable is chiral axis. So at what temperature it will start racemizing. So that's what I did then. So what I did is I took a pure diastereomer of the ligand and dissolved it in toluene for 40 hours and heated at different temperature. So as you can see there, as you increase the temperature, it will start getting racemized or epimerized, you can say. Okay, so as you can see that the DR was getting lower and lower. So you can see you can start with 99.8% of DR. As you keep increase the temperature, it will drop your DR. So maximum it will go up to 70-30 and it stops there. So that's what like 3:1, which we have seen during the synthesis as well. So then we calculated the barrier to rotation energy, which is required to rotate your chiral axis, and it was found to be 112 kilojoule per mole or 27 kilocalorie per mole. So then once we have ligand in hand, the next step was to apply it in reaction, right? So that's the main goal, to synthesize. That's why we synthesize the ligand. So we chose the reaction called A3 coupling. So what is A3 coupling actually? So A3 coupling is the reaction between aldehyde, amine, and alkyne. So you have 3A, so A3, right? So it is a copper catalyzed reaction. So you don't really require ligand over there. So if you don't use chiral ligand, so and if you use only copper metal, so what you get is you get a racemic propargyl amine. So propargyl amine, you have a triple bond over here, and you have here, so that's why it is a propargyl amine. Why we chose actually A3 coupling? So because you can generate this chiral amines, which are very, very important. So these are some of the drug molecules which has chiral amines into them. These are some of the very, very important chiral amines in academia as well as industry, and that's why A3 coupling is one of the easiest or straightforward reaction wherein you generate chiral amines, and that's the reason we chose A3 coupling for our study. Also, you can also synthesize a variety of natural products by using A3 coupling. So these are few of the examples wherein A3 coupling can be utilized during their synthesis. these are the couple of reasons we chose, like, due to which we chose A3 coupling. So okay, so the first thing first. So we then made a chiral catalyst. So how did we make chiral catalyst? So we use copper salt. Okay, various copper salts were there, but we found that, okay, copper bromide is the best among. So we then reacted 5 mole% of copper bromide with 6 mole% of ligand. So you will have to always use little bit excess of ligand so that there will not be copper remaining there, which can simultaneously form the racemic product. So therefore, like, yeah, you will need to always make sure you are using excess of ligand. So when we, and then react it together for half an hour, and then you add your other starting materials. So in this case, you use this aldehyde, aliphatic aldehyde, then you have this dibenzylamine and TMS acetylene as an alkyne source, in toluene as a solvent. So you generate propargyl amine in 52% yield and 93% initial selectivity. So this was the first reaction wherein we got the hit. So this is not always the case. You start with like 10%, 20% initial selectivity, and slowly, then you build upon it. You can optimize your reaction by changing various stuff. You change the copper salt, you change the solvent, you modify ligands. There are so many things, as I mentioned, but we were fortunate enough to get the 93% of initial selectivity in our first hit itself. So, but like, yield was not that great, and initial selectivity also can be improved. So we needed further optimization. So the further optimization was done. So as you can see, we changed alkyne bit. We changed the equivalent of alkyne. We increased from 1 to 1.5. Then we changed the temperature from room temperature to, we cooled it down. So generally in asymmetric catalysis, if you slow down the reaction or if you cool down the reaction, initial selectivity will enhance. The reason being is what happens as if you cool down the reaction is you slow down the reaction, right? So you slow down the reactions means you have, so if you slow down the reaction, there is a, if you slow down, there is a thing that if you slow down the reaction, you are giving a time, for chiral catalyst and your substrate to react, properly. And that's the reason you generally see this is the trend you generally see. When you reduce down the temperature, the E will go up. Okay, so we also changed the solvents. We also added the additives like molecular sieves. So as you can see that yield was slowly improving, and the E was also improving. And this was our optimized reaction condition, wherein we use 1.5 equivalent of this alkyne over here at 0 degrees centigrade in toluene as a solvent. By using molecular sieves, you generate this product in 89% yield with 98% of initial selectivity. So what's the role of molecular sieves here? So many of you might know that molecular sieves will act as a water quencher. So if you see this reaction, what actually happens here? So first of all, the aldehyde will react with amine, okay? And you get a water as a byproduct, and you generate iminium species, okay? So iminium species, and you get a water as a byproduct. So if you don't have anything which is taking care of your water, so if water comes out, so that's the reversible reaction. See, if there is a water availability, it will again break your iminium ion formation, okay? So if you have water over there, so if you have something which is taking care of your water, so you don't have that reversible reaction going on. And that's why you see the improvement of yield over here, okay? So once you have that iminium ion formation, so you generate the copper acetylide with the copper. So what's the role of your copper? So copper will generate the copper acetylide, and that's how then so your iminium species is electrophilic in nature, your copper acetylide is nucleophilic in nature. If you are nucleophile, what happens is you get a reaction, okay? So yeah, that's that. So then we started studying, various different aldehydes. So as you can see here, you have this isobutyryl aldehyde over here. So you generate an 89% yield, 98% E. Then when you change it to cyclohexane aldehyde, again yield can be improved further to 90%, and the E was found to be 97.5%. With the cyclopentene aldehyde, the yield was reduced a bit to 70%, but the E was close to 93%. With the smaller cyclopropane ring, the yield was still 65%, but E was drastically reduced, which is expected, which was kind of expected. You can because this is the cyclopropane moiety is quite small, and it will not have that much steric effect during the transition state, and that's why you see the reduced enantiomer selectivity. So you can see this then the other aldehydes wherein you don't have the alpha substitution, okay, still you get the good enantiomer selectivity, good yield. So then this hydrocinnamaldehyde, again, nice yield, nice enantiomer selectivity. Then you have this long chain aldehyde. Here also you generate product in 86% yield and enantiomer selectivity of 94%. Again, furthermore, alkyl chain, no problem. You can use that as well, and you can generate product in 66% of yield and 93.6% of enantiomer selectivity. What about the unsaturated aldehydes? So until now we saw aliphatic aldehydes, right? So when you use the benzaldehyde, so yield was drastically reduced and the E was also dropped. So the reason is benzaldehyde, the reaction between the benzaldehyde and the dibenzylamine, it's quite sluggish. So you don't get the iminium ion formation as good or as fast as your aliphatic aldehyde, because that's what the reactivity difference between your aliphatic and aromatic aldehydes. So again, you can see the similar trend with the cinnamaldehyde and this propargyl based aldehyde. So E was quite less, but in upcoming slide, I'll show what I did and how did I improve the enantiomer selectivity for this substrate up to like 98-99%. So I'll talk about this in the upcoming slides. So again, by changing the TMS acetylene to the phenyl-acetylene, no problem. You can generate the product in 95% yield, and you can get E up to 80%, which is not bad. But for some reason with the cyclohexylaldehyde, E was dropped to 38%, but yield was quite great. So yeah, so if you see here, so in earlier slides, okay, so we use 5 mole% of copper bromide and 6 mole% of ligand. So normally we do reaction on 0.2 millimole scale, and the best yield we got for this particular substrate was 89% and 98% of E. So, but we said, okay, we can increase the scale of the reaction by 10 times and decrease the amount of copper bromide and ligand to 1 mole%. Okay, so we did that. So we use 1 mole% of copper bromide as a copper salt and 1.2 mole% of the ligand, which was made by us. And yeah, actually it should be 2 millimole. So yeah, so that's the mistake there. So yeah, in this case also you can generate the propargyl amine in 95% yield, and enantiomer selectivity was also 96%. There was a drop in enantiomer selectivity, but that's not much actually, just by 2%, which is quite acceptable. Then what we did is we know that we can make diastereomeric mixture of ligand. So we thought, okay, let's see the effect of other diastereomer, what happens. Okay, so we made the mixture of the ligand which has a different diastereomeric ratio, and we applied that in a reaction and see the effect. Okay, so as you can see that if you have the less DR, so you get a less E, which is the trend. Okay, but in some cases it is seen that even though you have a other enantiomer or diastereomer of your ligand, the major, only one enantiomer or diastereomer will act as a catalyst, and other will just sit and will not do anything. So that's how, like, that is seen actually in quite a few reactions. So we thought let's see what happens in our reaction, but we didn't see anything like that. So which wasn't surprising. We just wanted to see what happens. And yeah, so this is the trend. So you see the product E will reduce down as the D of the ligand goes down. So if you see the ligand structure of the ligand, you have a chiral axis, you have this imidazoline ring, and also you have this phosphine moiety. So I wanted to see how, like, what's the importance of each thing, in the ligand. So how important is the imidazoline moiety? How important is this diphenyl phosphine, or how important is the chiral axis? Because we have chiral axis as well as chiral center, we didn't know like whether it's the chiral axis which is helping to generate enantiomer chiral product, or is it the central chirality. So what we did is we made a ligand without axial chirality, right? So you don't have this extra naphthalene ring, and you don't have this electron withdrawing pentafluoro of phenyl groups. So you don't, if you don't have that, you don't have the axial chirality. And that's what we plan to make this ligand, this new ligand, wherein you don't have the axial chirality. So, and I also made this ligand wherein you don't have this extra nitrogen, you just have the oxygen. And we wanted to see like how it will affect during the catalysis. So what happens, like if you see the difference between these two is, so this nitrogen is a bit more nucleophilic as compared to this, because of the nitrogen lone pair over here. So yeah, we wanted to see the difference between those two, how it will affect the reaction. And in the third ligand, wherein you don't have the diphenyl phosphine, instead you have the nitrogen only. So yeah, that was the plan. So we synthesize this ligand which doesn't have the axial chirality by starting with this 2-bromobenzaldehyde and this chiral diamine in presence of magnesium sulfate, which will act as a water quencher over there, and our earlier reagent, which is N-bromosuccinamide in DCM as a solvent. So you get this imidazoline ring, bromoimidazoline in 54% yield. Then you use sodium hydride, which will act as a base. It will take this proton and you generate the anion, which will attack onto your benzyl bromide in SN2 fashion. And you get this N-benzylated bromobenzene in 77% yield. And then you do the final cross coupling between this aryl bromide and diphenyl phosphine using copper catalysis in presence of base in toluene at 100 degrees centigrade. So you generate this CP bond over there in 73% yield. So then it was time to apply it in a catalysis. So as you can see here, we did that. And this was not expected actually. We could see still the, 83% of E, but yield was reduced little bit. So this was at room temperature. So if you do the reaction at room temperature, E will not reduce, but reaction will be a bit faster, and you can improve the yield of the reaction up to 72%, and E was 83%. But the most important thing is, in earlier case, it was the S enantiomer, and in this case, we saw R enantiomer. So you can see in earlier case, this N-benzyl moiety was backside, and here it is towards you, right? So it is quite surprising actually to know that. So you can generate, like whichever enantiomer you want. So that's easy. So then we wanted to see the oxazoline, how it behaves. So we also made this by using the route shown over here. So then we started with this amino alcohol, which is enantiomolecularly pure, and reacted with this 2-bromo benzoyl chloride. You react specifically or selectively with this amine. So you have a base, it will take this proton over here, and then you get a reaction with this acyl benzoyl chloride here. You generate this amide, and then in next step you take you use triethylamine and acetyl chloride. So triethylamine will act as a base. It will take proton from here, your hydroxy, and the acetyl chloride is your this, it will displace this chloride and you'll get this OMS. So OMS is very good leaving group. So when you react this amide with sodium hydroxide in methanol, so you generate this oxazolene. So the mechanism is like, first, what sodium hydroxide does, it is a very good base, right? So it will, so if you see this molecule, there is only one hydrogen. So it will take up this hydrogen and you generate that anion, and then it will like isomerize, delocalize its electron, and you generate this O minus there, and it will displace your OMS. Now you see the enantiomer selectivity. So sorry, you see the chirality over here. So it has changed, right? So you started with this phenyl being wedge. So now here it is behind, right? It's not in the front. So why that happened? So you know that in your SN2 reaction, you see the inversion of chiral center, and that's what has happened there. Okay. So, and then you do now normal your copper catalysis by using diphenylphosphine and generate the ligand in 58% yield. Then again used in the reaction, and there was no reaction at all. So now you can see just slight change in the structure, you completely stop the reaction. So that is what asymmetric catalysis is. You don't know what is going to work. Okay, so there are a lot of efforts. It's just like a trial and error. You just keep trying. So normally we would have seen, like we would have expected, okay, we might get some percentage of PE, but you see nothing. So then, yeah, we also synthesize the third ligand. Similarly, like this NIS, this time we use NIS. We felt like NBS wasn't as good as NIS, so it is like N-iodosuccinamide. So you generate, sorry, am I taking a lot of time? I don't know, what's the time?
Please continue, sir. No problem.
Okay, yeah, I'll finish in soon. Okay.
Yes.
So we generated this diphenyl, this ligand. So you can see the crystal structure. No reaction this time. You can see how important the phosphine was. Oops. So a bit fast. I think I took a lot of time. So yeah, this is the, in conclusion, you can see that the main ligand furnish the S enantiomer, the other ligand which doesn't have the axial chirality furnish the R enantiomer, and the third and fourth ligand, yeah, it didn't furnish anything. So we published this in the journal called ACS Catalysis in 2017. And also we wrote a review article in ACS Catalysis in 2018 about the PN ligand. And we also wrote a book charter in World Scientific. That's that. Then I was talking about the aldehyde, benzaldehyde wasn't reacting. So we changed a couple of things. We changed, the alkyne moiety to up to this. And you can see there E wasn't improved. Then we changed amines. As you can see here with the cyclic amine, the E was 97% and yield was 94%. So it was quite good. So we then started reacting this aromatic aldehydes with secondary amines and your alkyne. As you can see in all the cases, yield and E was very good. So these are the ortho substituted aldehydes. Then you have meta substituted. Here also no problem. You can see up to 99% of E. Then few more like para substituted aldehydes, no problem. You can use various miscellaneous aldehydes, no problem at all. Good yields. Also like here various heterocycles, no problem at all. And also then now you can see that we took a X-ray crystal of the product. Herein you can see the hydrogen is at the back and your this heterocycle was front. So yeah, we also use the cinemaldehyde and propanaldehyde successfully, no problem like earlier case. These are some different cyclic amines, no problem. You can use all of them. yeah, you can use this alcohol as well, no issue at all. We also did similar stuff like earlier, and here we can see that we got the E up to 99% without axial chirality, so both ligand work as good as, and also here also we got the different enantiomer as compared to our original ligand. So we use a product to generate different compounds, so we can synthesize, this terminal olefin or this allenes from the product we generated. This was published in 2019 in Journal of Organic Chemistry. We also reported this a review actually in 2019, ChemSoc Rare, and also we wrote this chemical review on oxazolone-based ligands. In acknowledgement, I would like to thank my research supervisor, the ex-supervisor, Professor Pat Gairy, IRIS Research Council, SFI, and Biomedical for funding. And finally, I would like to thank you for the invitation and listening to my talk. Thanks.
Thank you,
Cheers.
Dr. Balaji, sir.
Sorry, did I take a lot of time? I'm sorry about that.
No, it's a very inspiring to the students. Those are from the rural area, and they are going to be the PG in the next year, and that will be inspiring to the students, definitely.
Okay, thank you.
And now the session is open for the discussion, and may I have a few questions from the audience. Those have been there. Please, anybody's there? I welcome those good questions. Please, can we have the questions? Yes, please. Okay, if there is no questions, then let's have a big claps for the from the online mode for this good presentation from Dr. Balaji Ropre sir. Is a scientist from Ireland, and he has presented a lot of his research, postdoctoral research work on the topic of particularly asymmetric catalysis. And at the beginning, he has discussed on the preliminary introduction of these asymmetric catalysis, and then he's stepped over to the chiral ligands, axially chiral ligands, particularly as a catalyst, and he's go through the development of chiral ligands, and then he's discussed on the particularly which was his completed in the 1 year research work, and he's particularly developed a single crystal array, then a lot of NMR, IR, and then he concluded the application of asymmetric chiral ligands as a catalyst for the synthesis of the various drug molecules, such as the cetirizine, which is a very, very important drug molecule for the common beings. Those are the people who may suffer from the various, say, for example, we will say it is. Okay, then also he discussed the scope of A3 coupling for the aldehydes and acid molecules, where he focused on the retrosynthesis of the various compounds, and finally he discussed focused on the specifically on substrate scope, particularly in the A3 coupling. So this is a very good research work he has carried during his postdoctoral research work at various research institutes, and because of that he has a very good research publications in like the Journal of Organic Chemistry, which is the ACS, American Chemical Society Journal, then RSA Advances publications. So these are the big publications what he has got through his very good research. So we would like to thanks Dr. Balaji Ropre sir for his best presentation. We have really inspired with your presentation, and I definitely assure you that my students, those are from the rural area like Sangola, they may get definitely enlightened with your presentations. And I also hope that these are the students, those are going to be post graduate study. And I know that from today's morning session that the Sangola, at Vidyan Mahavidyalaya Sangola will starting their PG course in organic chemistry. And so I definitely say that this is the very, very inspiring lecture for these BS students, and definitely they go for the PG at their Vidyan Mahavidyalaya Sangola College. So from the organizing committee of this today's conference, I would like to play gratitude for you, and thanks for many for this presentation. Thank you, sir.
Thank you. You're welcome.
Yes.
Thank you, sir. Thank you, Professor S.H. Gaikwad, sir, for your valuable discussion and handling the session third. Also, my sincere thanks goes to Dr. Balaji Rokde, sir, for your wonderful presentation. Now we will start session fourth. Once again, I welcome all in session fourth. The session coordinator is Mr. A.R. Ingole, sir. I request Mr. A.R. Ingole, sir, please. Ingole, sir.
Thank you, sir. Here I would like to introduce for the Professor Dr. Lalasaheb Kashi, chairperson of this session. Professor Kashi obtained his BSc degree, securing first class with distinction in the year of 1988 from Shivaji University, Kolhapur, and MSc Physical Chemistry with first class in 1990. And he also got a MPhil degree with A grade in the year of 1994 from the same university. He has acquired his PhD degree in 2012 from Savitribai Phule University, Pune. Professor Lalasaheb Kashi has made a significant research contributions in the field of electrochemistry, especially proteometry and the pesticides. He has published several papers in national and international journals. Dr. Kashi has worked as a teacher, as a researcher, and as an academic administrator in a professional career of about 28 years. He has started his career as an associate professor in chemistry at Sangameshwar College, Solapur in 1990. And then he has been appointed as head of Department of Chemical Science at Vidya Pratishthan's Arts, Science and Commerce College, Baramati from July 1995. Now he is working as a vice principal since from 1914. Now under his leadership, he has been started PG course in analytical chemistry and PhD research center in chemistry affiliated to Savitribai Phule University, Pune. So this is a brief introduction about Professor Dr. Lalasaheb Kashi. Now I heartily welcomes and request to Dr. Kashi, please take the charge of this session. Thank you.
Hello, am I audible?
Yes, sir, it's audible.
Yes, okay. Thank you, Professor Ingole, for brief introduction. First of all, I congratulate organizer, Department of Chemistry, Vidyan Mahavidyalaya, Sangola, India, for organizing such wonderful international e-conference activity on proteomic application to biomedical research. Welcome to today's fourth session. There are two oral presentations. First from PVPM College, Tasagaon, India, Dr. Ajay Ambure, and second is from Dr. D.Y. Patil, Women's College, Pimpri Chinchwad, Pune, Dr. Sunil Gaikwad.
Participants, authors are requested to present your content within 8 minutes, followed by discussion. Now, I request Dr. Ajay Ambure to present your research work. Thank you. Dr. Ambure.
Thank you, sir. Thank you very much. Am I audible, sir?
Yes, yes.
Yes, yes.
It's all right.
Thank you, sir. So here I am. Sir, the screen share is successfully.
Yes, it is visible.
Okay, thank you, sir. Thank you very much, sir. Firstly, I am thankful to Vignan Mahavidyalaya Sangola for inviting me for the oral presentation in the International E-Conference on Proteomic applications of biomedical research. Myself, Dr. Ajay Ambure, as an assistant professor from the Department of Chemistry, Padma Bhushan, Dr. Vasantrao Dada Patil Mahavidyalaya, Tasgaon, District Sangli. And today I am here to present my research work on design synthesis and evaluation of pyridine 4-il-134 oxadiazole 2-il-thioethylidene hydrazine carbothiamide derivative as a potent antitubercular agent. So before starting our research work, here we discuss about the heterocyclic compound. As we know, the heterocyclic compound showing the large spectrum biological spectrum, that is the biological activities like the antitubercular, antidiabetics, anti-cancers. Number of bioactivities are shown by the heterocyclic scaffolds. That's why researchers are continuously work to synthesize the novel component, the novel derivatives having the heterocyclic scaffold. And here also we try to find out such type of heterocyclic scaffolds showing the biological activity. Now in this work, during the synthesis, the researchers nowadays follow the protocol of green chemistry to find out the eco-friendly route for the synthesis of any derivatives. Here also we find we use the two main cores for the synthesis of our novel compound, and that two main points, they are the PG400 as a green solvent and BEC, bleaching earth clay, as a heterogeneous catalyst. So the PG400 advantages on the traditional synthetic route, that is the recyclability, simplicity in handling, easy isolation, acts as a phase transfer catalyst, easy transformation, non-toxicity, as well as the bleaching earth clays having the pH 12.5 used as a heterogeneous catalyst, which shows the advantages like recyclability, simplicity in handling, catalysis, isolation, minimum execution time, low corrosion, easy transport, non-toxicity, and waste minimization. That means we followed some of the principle of green chemistry for the synthesis of the new heterocyclic scaffold. Now, I am here to present the design, synthesis, and evolution of pyridine scaffold embedded with the hydrazine carbothiamide derivatives with oxadiazole. Now, when we design such type of heterocyclic compound, we focus on the specific bioactivity, that is the antitubercular. Anti-tuberculosis is the leading cause of death in the world and largest burden on the developing countries. Tuberculosis is one of the present disease caused by a pathogen Mycobacterium tuberculosis. Now, the main drawback during the treatment of tuberculosis that the TB necessarily required long-term reasons due to the non-replicating parasite tuberculosis parasite. It is found that the improper treatment generate a harmful effect and make a significant contribution to emerging of drug resistance strain. This appears and impressive to combat the increasingly frequent drug resistance strain on Mycobacterium tuberculosis. On the basis of that, and finding by the different literature, we find out that the hydrogen carbothamide is one of the best scaffold which shows the number of biological activities. But beside that biological activities, it shows the biological activities against the Mycobacterium tuberculosis. That means it also acts as a TB drug. And with this hydrogen carbothamide, we also find out that the oxadiazole is also an important scaffold from the heterocyclic compound, showing the different biological activities like anti-cancer, anti-convergent, anti-HIV, anti-inflammatory. On the basis of that, here we embedded the hydrogen carbothamide derivative with this oxadiazoles to form such type of a molecule which showing bioactivity against the multi-drug resistance bacterium, the Mycobacterium tuberculosis. So in this design, during the design of that molecule, we use this oxadiazole moiety with the thiocemic carbazide. Now the oxadiazole moiety is also attached with a specific core of the heterocyclic compound, which is having an antibacterial activities. That means this template for the anti-tubercular moiety as well as the fragment from the drug unit and the thioacetazone as an active pharmacophore. Such design we try to synthesize by using the green route. Now, in this synthetic route, actually this reaction goes through the 3 steps. For conveniency and due to the time, here the first 2 steps here we presented in one scheme. That is the synthesis of 1-phenyl 2,5-pyridine 4-yle 1,3,4 oxadiazole to yle thioethane 1,1 derivatives. Now during this 2 steps, first step is formation. In the first step, that reacts with CS2 in presence of KOH for 3 hours to form the compound second, which again reacts with substituted phenyl bromide to get the compound 4A. Now these are the common reaction which is we are found in a number of literature, and that means the known reaction of formation of 4A to K. But here also we synthesize these 2 components, and it can be characterized by spectroscopic technique by IR, NMR, as well as C13. On the basis of which we find out we confirm the formation of 4A to K. Now the main route or main step for the synthesis final compound is this third step, where the compound which is formed by the condensation of compound 2 with substituted phenyl bromide can be reacts with thiazide carbazole in presence of PEG 400 and acetic acid uh, up to 70 to 80 degrees centigrade for 2 to 3 hours stirring, we get the final compound, that is the pyridine 4-eel-134 oxidazole 2-eel thioethylene hydrogen carbothiamide derivative. This formation of hydrogen carbothiamide derivatives also can be characterized by using the IR, by using H1 NMR, by using the C13 and mass spectroscopy. Specifically, the formation of the compound can be confirmed by the absence of the carbonyl group, as well as formation of the primary and secondary amine in the IR spectroscopy, IR spectra, as well as the in the NMR spectra. NMR spectra, all these four aromatic peaks, as well as these two, all these four aromatic peaks, they are present as usual in the H1 NMR spectra, but with this four, it gives the NH2 and NH peak specifically in the H1 NMR spectra, which confirm the formation of the final product, as well as the mass spectrum is also showing, it also gives the information about the confirmation of the final product. So after confirmation of that product, we use the biological study of the newly synthesized radionuclides by using the RIMA assay for the antitubercular uh, for the antitubercular activities against the reference like the rifampicin as well as isoniazide, because the isoniazide is one of the component from which here we synthesize that compound, and from this antitubercular activity, from this RIMA assay, when we find out the MIC of the compound, some of the compound shows the excellent MIC report. That means it gives the 3.90 to 7.80 milligram per ml MICs as compared to the rifampicin as well as isoniazide, which showing 0.24 to 0.48 microgram MICs. Now, this is about the MIC values, which can be confirmed by molecular docking study. The molecular docking study, or the binding energy of the newly synthesized compounds with the specific binder force. That means these molecular docking studies can be confirmed by the pharma mapper survey, and binding with the CYP51 ligand confirmation for the antitubercular activities of the newly synthesized compounds. So this is the molecular docking studies which show the binding form of the compound which is synthesized during this route. And the conclusion from this synthesis, that the study of relevant and stability of newly synthesized pyridine 4-eel-134 oxidazole 2-eel thioethylene hydrogen carbothiamide derivatives as a potent antitubercular agent. The SR study linked the requirement of bromo, nitro, and methylene substituent. That means the three substituent, which is called as a key substituent from the manifesting for the antitubercular activity. This SAR includes the electron withdrawing substitution showing the high activity of that compound. The present investigation demonstrates the successful contribution to the ligand-based pharmacophore model for the inhibition of CYP51 in mycobacteria, which confirm the antitubercular activity of the synthesized compound. Binding free energy value indicates that the compound from the present study can be potentially used to lead the compound for further developing the effective CYP51 inhibitor. Based on in vitro observation and the light of structural analysis of docking complex, we tried to propose the possible mechanism of action of pyridine 4-eel-134 oxidazole 2-eel thioethylene hydrogen carbothiamide derivative in Mycobacterium CYP 51 inhibition. Okay, so this is a short synthesis which is doing with my friends, and I'm here also happy to knowing that this research work is published in computational biological and chemistry in the Elsevier journal in 2019. So this is all about today's presentation, and due to short time as well as uh, after the that means yesterday I got the invitation for the presentation, and in one day I completed my presentation for my research work. So thank you, sir. Thank you very much.
Thank you, Dr. Ajay, for your wonderful presentation on design, synthesis, and evolution of pyridine oxydiazole thioethylidene hydrazine carbothiamide derivative as a potent and anti-tubercular agent. Now, presentation is open for discussion, or you can ask any questions from online audience, online participant. Any question? Okay, sir. Thank you.
Okay, sir. Thank you, sir. Thank you very
Now,
much.
I request Dr. Sunil Gaikwad from D.Y. Patil Women's College, Pimpri Chinchwad, India.
Yes, sir.
Dr. Gaikwad.
Yes, sir. I'm sharing my screen, sir. My screens are visible to all of you.
Sir, screen is visible, but you should start from first slide.
Last slide.
Yes,
Where is
sir.
the first slide?
Yeah, this is my first slide.
Click on first slide.
Yeah. Okay. Now,
Take first slide first.
yeah. Now I'm on the first slide only.
You can scroll. Yeah.
This is first slide, right? Now it is on the first slide. Yes, sir.
Yes, sir.
Okay, thank you very much. Good evening to all of you. I'm Dr. Sunil Gaikwad. I'm assistant professor working as ADO position, Department of Chemistry, Dr. D.Y. Patil, ACS College, Pimpri, Pune. Initially, I would like to thanks the Vigyan Mahavidyalaya Sangula for arranging this wonderful international e-conference on the proteomics biomedical research. Here, I'm going to present on the molecular iodine as a versatile reagent, a synthesis of the bioactive compound and synthesis of the lithophilic acid derivatives. Here, as we know that how important is the beta carotene derivatives. We know that the beta carotene derivative is nothing but a pyridoindols tricyclic molecules, and this beta carotene easily available in the edible and the non-edible foods. The edibles means the tomato, orange is there is, and non-edible is the sarpaganda, and there are other naturally occurring fruits and oils also that available a beta carbolene. Okay, and the beta carbolene has a very good properties, such as they act as a anti-cancer, anti-malarial. They generally use as anti-malarial and anti-cancer in the older era. In the China, they use a Harmeran as an anti-malarial as a drug candidate. And presently, as we know that the tadalafil, one of the good drugs which is available in the market as a, for the neurological treatment. Here, the iodine DMSO catalyze chemoselective biomimetic aromatization of the tetrahydro beta carbolene 3-carboxylic acid and mechanism study by the DFT calculation. As we know that there are various methods available for the aromatization of the tetrahydrobeta carbonyls to the beta carbonyls. As you can see in the left hand side, there are various reagents available for the aromatization of this tetrahydrobeta carbonyl to the beta carbonyl. But the thing is that they are all these reagents aromatize this tetrahydrobeta carbonyl 3-carboxylic acid to the beta carbonyl, having a decarboxylation of the acid group. Here the process which is carried out by aromatization with the intact of the acid group, which is carried out by the enzyme that is E. coli MCPP. Here we have developed a iodine DMSO H plus at 60 to 75 degrees Celsius up to the 1 hour. When we treated this tetrahydrobeta carbonyl in iodine DMSO H plus, we'll get the aromatic beta carbonyl without decarboxylation of the acid group at only 60 to 70 degrees Celsius, and up to the reaction we have to heat it up to the 1 hour only. If we heated the reaction more than the 1 hour, then there is a chance to decarboxylation of this C3 acid group. And here the developed methodology we have applied for the series of the beta carbonyl derivatives, and we synthesize this series of the beta carbonyl derivatives as this we developed a methodology, and the mechanism was also studied by the DFT calculation, and we found that the energy of this our desired product is a minimum that the decarboxylation product.
Yes, sir.
So, yes, sir.
You have to change slide.
Yes, sir. Yes.
Check slide. It is on first slide.
Yeah, I am on the first slide only, sir. This. Okay. Now. Hello.
Even it is the first slide. Go
Yeah,
to second slide.
Yeah, it is the first slide only. Now, here again we have developed a new method for the aromatization of the tetrahydrobeta carbonyl, that is chemoselective dehydrogenation of the tetrahydrobeta carbonyl to the beta carbonyl. Here we have treated this iodine DMSO in H2O2 hydrogen peroxide at 60 degrees Celsius, and we got the 3,4 dihydrobeta carbonyl with the 80 to 92% yield. When we heated that the same tetrahydrobeta carbonyl the above 90 to 100 degrees Celsius, then there is complete aromatization of the beta carbonyl to the beta carbonyl. And we have here the developed methodology we used for the synthesis of the. Yes, sir.
Sir, please.
Yes,
Your current slide is not visible.
Current slide is not visible. Now, sir.
Yeah.
Now
Okay.
it is visible.
Yeah.
Here we are the developed methodology we applied for the synthesis of the series of the 3,4 dihydrobeta carbonyl, and we have synthesized the series of the 3,4 dihydrobeta carbonyls, and as well as the complete aromatized the beta carbonyls at 100 degrees Celsius. The developed methodology we have utilized for the synthesis of the natural product eudistumin U, eudistumin N, harmanin and non-harmanin derivative, and coumagen C as one of the very good natural product. Here we have applied the iodine DMSO for the aromatization of the beta carbonyl with allylation and the deallylation processes. As we know that the allylation and the deallylation, or the protection and the deprotection, is a very good for the organic transformation. Here we have treated the iodine DMSO in H plus and iodine DMSO in H2O2. What we found that when we treated this iodine DMSO in H2O2 region with O allele protected tetrahydrobeta carbonyl, then what we get, there is aromatization, but there is no deallylation of the product. When we treated the same O allele substituted tetrahydrobeta carbonyl with H2O2 iodine DMSO H plus, there is aromatization as well as a deallylation takes place. The mechanism of on the basis of this result, we found that the mechanism goes to the, when we use the iodine DMSO H2O2, the mechanism goes via the free radical mechanism. Because of that, there is a no deallylation, but there is a aromatization. While using the iodine DMSO H plus, the mechanism goes to the aromatization, but there is a deallylation. That is the mechanism goes the general mechanism, that is the generation of the in situ HI. The next is design and synthesis of the novel lithocholic acid derivatives as a potent anti-cancer inhibitor. As we know that the how lithocholic acid is important for the metabolite, and this lithocholic acid is important for the to triggers the transcriptor activity of the vitamin D. And we know that how important is the vitamin D. The vitamin D is used for the cell growth, calcium reabsorption, bone calcium resorption, VDR sensor. And we know that why we didn't eat the much more vitamin D, because as we know that vitamin D is also act as an anti-cancer agent, but we cannot eat the much more vitamin D because of it gives the calciumic effect. It deposits the calcium or it weakens the calcium in our body in our older age or in the younger age also. Because of that, we cannot eat the higher vitamin D tablet is. Our doctor also suggests that the don't eat more than the vitamin D tablet, more than this 4 or 6 vitamin D, right? Because of it deposits the calcium in our body, or it gives the calciumic effect. Because of that, we cannot eat the much more the vitamin D. Because of that, as we know that this lithocholic acid derivative is triggers the transcriptor activity of the vitamin D, number one. And second one is it shows the non-calciumic activity, non-calciumic activity. Because of that, we did the molecular docking study with the 1DB1 protein with the modified lithocholic acid derivatives, and we found that when we modification text did at this A ring with the hydroxy group or tertiary hydroxide with a different ester group, and about some molecule we selected which showing the very good docking result in 1DB1 protein, and out of that we synthesized some of the best docking score molecules. And one of that, this is the 5A, that is a tertiary group, tertiary hydroxy group attached at this A ring. We started this synthesis from the normal lithocholic acid derivatives. Initially, this acid we reduced to the alcohol, that is a primary alcohol. Then in the structure A1, we have the 2 hydroxy group. Then the chemoselectivity protected by only one primary alcohol group by using the TBSCl, tertiary butyl silyl chloride, and we will got this primary hydroxy group protected lithocholic acid derivative. Then the secondary hydroxy group then again oxidized by using the PDC, and we got this ketone molecules. That ketone molecules undergo Wieman's Horner-Wittig reaction, and we got this alpha beta unsaturated ester. Here is the interesting thing that we got the mixture of the E and Z, that is equal mixture. That is the E is a 50%, E is a 47%, and Z also the 46 to 47%. And then that isomer we separated by using the 0.5% of the ethyl acetate by using the HPLC. Again, this alpha beta unsaturated ester we reduced by using the H2PDC. We got this beta isomer of this ester, and that beta isomer ester then treated with the methyl magnesium bromide, that is a Grignard reagent, and we got this tertiary alcohol. And we know that this initially we have protected this TBSCl primary hydroxy group by using the TBSCl. Then that hydroxy group again deprotected by using the tertiary butyl ammonium chloride, and we got this primary hydroxy group. Then this primary hydroxy group again oxidized by using the PDC, and again this aldehyde oxidized by using the oxone. And the work and we got this equatorial position hydroxy group. And here also we synthesize the other isomers also by using the same methodology. And here we have synthesized that 10A compound having the stereochemistry at the axial position. These are the alpha isomer, and these are the beta isomer. Here the beta isomer we have synthesized. Initially this alpha beta unsaturated ester we have reduced asymmetric reduction by using the copper chloride, and here we have used S-tolbinab. We have used R-tolbinab, the chiral asymmetric chiral reagent. Here we use the S-tolbinab. Only S-tolbinab gives the axial stereochemistry, asymmetric reduced axial stereochemistry of this ester moiety. And again, we have this primary hydroxy group deprotected by using the HF. Here we have used HF because in the asymmetric hydroxylation reaction, we have used polyhydroxymethylsilane, and this polyhydroxymethylsilane is a sticky compound and it bound to the compound and it remain as it is with our product. Therefore, we have used a strong acidic condition to remove this polyhydroxymethylsilane, and the primary alcohol then oxidized by using the PDC, and we got the aldehyde, and this aldehyde again oxidized to the carboxylic acid. And the stereochemistry of this axial is confirmed by using the X-ray crystallography, as well as this E and Z isomer of the alpha beta unsaturated compound is also confirmed by the X-ray crystallography. And all this synthesized compound, we have synthesized 5 compound, and out of this 5 compound, only 1 compound shows a very good BDR receptor recombinant activity with the vitamin D. And all this work is published in the bioorganic chemistry journal, and they have reported, one is a 2 patent, one is a Spanish patent, and one is a WIPO patent, word intelligent property the patent. The second, I would like to thanks for the organizing committee for giving me such wonderful opportunity to present my some of the research work in your international e-conference. Thank you very much, sir.
Thank you, Dr. Gaikwad, for presenting such a wonderful your research work on molecular iodine is a versatile reagent synthesis of bioactive compound and synthesis of lithocholic acid derivative. Now, the presentation is open for discussion, or you can ask any question from online audience. From the student side, okay. Dr. Gaikwad,
yes, sir.
What are the techniques we are using for the identification and interpretation of all these compounds in your research work?
Generally, we initially used H1C13, and finally the mass of the compound was characterized
because the
HRMS.
molecules are complicated. You can hear.
Yes, sir. All the molecules are not complicated, that much complicated, or the symmetrical molecules. They have having the carbon. Initially, we characterized by using H1C13, and all the masses were confirmed by taking the HRMS, because we got the exact molecular formula of that particular compounds. And all this work we have published in the chemistry select. One is a tetrahydrolator, and this one is published in the heterocyclic chemistry, and the other part this other work is published in the bioorganic chemistry. Here we have reported 2 papers, one in bioorganic chemistry, all the different molecules.
Okay, thank you. Now over to Professor Indole. Thank you.
Thank you, sir.
Now, time comes to express my sincere vote of thanks for this session. Here, we are sincerely thankful to Dr. Lalasaheb Kashi for giving his valuable time for our e-conference and conducting the oral discussion section. Our institution, Department of Chemistry, and on the behalf of all participants, we are very thankful to Dr. Kashi. We are also thankful to Dr. Ajay Ambure as well as Dr. Sunil Gaikwad for presenting the research papers in our e-conference. Now, overall, we are thankful to all of you. Thank you.
Thank you.
Thank you, sir. I also thanks to all the participants, organizers, technicians, colleagues, and non-teaching staff for their efforts. I declare that today's sessions are over. Let's meet tomorrow with same e-conference with same theme. Thank you very much. Have a good time. Thank you.
Thank you. Thank you.